Good morning. Please let me know in the chat if you are present for the day. I forgot my coffee somewhere. Let me find my coffee. Okay. Let's talk about what we're up to today. So we have, officially at this point, completed Unit 1. Hooray! We learned a little bit about the diversity of living things. Lots of different stuff out there. Some of you have started to hand in your projects already, which is nice. Remember that the due date is at midnight tonight. If you are heading towards that due date and you have not completed one of the 3 items, the vocabulary list, the major assignment, which is the species spotlight assignment for this unit, and the quiz, please send me some email, some communication. Okay, let me know what's up. If you feel like you're not going to make that deadline, and in your email, make sure that you're letting me know your proposed alternate deadline when you feel like you will be able to complete it. Okay, and then abide by that deadline. So anyway, it happens. I realize that this course moves extremely quickly. It's probably not that different from physics, or if you're taking another senior science. From what I've seen and discussed with my colleagues, I mean, it's a lot of content in any senior science course that we're kind of attempting to compress into this environment. But excuse me. But anyway, it's still a lot, so I'm not going to pretend it's not a lot. It's a lot. So please let me know if you're not going to be able to make the deadlines. You got to stay in contact. And that's just so we can work together to keep you on track and we don't end up with a situation at the end where you're not getting the credit. So I've had this exact same conversation with my other class. I'm teaching this course twice this quad. Unfortunately, there are already people in the other class that are way behind. Not a lot. No, that's very few, but they haven't contacted me. And so we are getting into trouble already. I've only been with you guys for about a week. And already I've got some people in my other course that I haven't seen for 5 days that are in trouble. And I hate to see it. It really bothers me because every time that's happened in previous quads, when a student has lost communication with me and has kind of gone off on their own and it hasn't worked out well. So just don't be that person. Don't be that person. I understand that the deadlines are tight and that there's actually quite a bit of work to do and that you'll have work in your other courses, which I also understand. But that's not an excuse not to communicate with me and let me know where you're at. So that's really the least you can do, is just be communicative and work with me to help keep you on track. If you're not willing to do that and not even communicate with me about the deadlines, I mean, like, really, what are your expectations of me? And I can't help you. It's most likely you're not going to help yourself in that situation, and then again, bad things happen. So I won't harp on this too much, but it bothers me so much when students don't get the credit at the end. I really want students to be successful in this course. It breaks my heart when it happens, and just because of just poor planning and things like that, things that you really have control over. So I've never had a student fail this course due to the content being too difficult, by the way. That's not to say that there aren't some challenging concepts in here. Diversity is fairly straightforward. We're going to talk about meiosis shortly. And yeah, it's a complex process. It is, some students have difficulty with it, 100%. But overall, I've never had a student that was unable to at least show me moderate understanding of the course content through the quizzes and the assignments and get the credit. And not everybody's finishing the course with 80s or 90s, certainly not, but everyone who is making a concerted effort is getting the credit. So just so you know, don't despair. I mean, a lot of you are probably not shooting for just get the credit, but And by the way, if your mark is somewhere where you don't like it, I realize you don't have any marks back yet, but when you do get them, start getting them back, if your marks are somewhere and you don't like where they are, there is lots of ability to change that. There are many assessments. There are 3 per unit. There's 5 units. So there's quite a few. I can discuss some quiz preparation strategies with you. I've done that with several students and it's really seems to have helped. We can discuss your assignments and sort of what is holding you back in terms of getting your assignments where you want them. Often it's not reading the instructions, by the way. So for those of you handing stuff in today, something to consider. Most of the marks that students lose is because they don't actually read the instructions and they leave stuff out that is a requirement of the assignment. So I would say that's the vast majority of places where students lose marks is just not reading the assignment. They're just kind of make an assumption about, do I need this or do I not need this? And if they're not sure, I mean, there's one easy way to find out. Just ask me. Like I'm happy to just tell you. So, but anyway, everybody has an opportunity here to be really successful if that's what they want, and I'll help them get there. So, you got to take advantage of it. All right, genetics, day 6. Well, wait a second. Do we have a piece of day 5 left over? No, we finished this. So right at the end of day 5, you would have completed that coursework. Ooh, I can't remember if I asked you to watch these videos. I meant to. Darn if I didn't. I meant to. Ideally, you watch this little summary video of cell organelles. If not, you can always do it at the beginning when you start working today, but it goes over what each of them does again. Does anybody have any questions about any of the cell organelle function stuff before we move on? Because I'm gonna call on it as we're discussing today's topic. Sorry, Bree. The screen is indeed black. No, it just says air server. Is it working now? Yeah. Okay, here we go. Sorry, these are the videos that I'm referring to here. So these are on the day 5 tab. There is a review of cell organelles. Again, if you didn't watch it yesterday, it's not a huge deal. Would probably not be a bad idea to watch it today if you didn't. What we're going to talk about today is this concept of transcription and translation. And these videos were meant to be something that you watched yesterday as an introduction to the process before I discuss it with you. Can I just put this out to the class? Actually, you know what? I'm going to try something right now. I'm going to try a new polling feature. Let's try this. Nice simple question. I've never done polls before, but I'm gonna try and incorporate this. How do I do this? Okay, so how it works is you click this link that's in the chat. Oh, or I could, a QR code. I like it. How do I do this? How do I show you the QR? I guess you could just click the link. You could just click the link. But I could show it to you. How do I do that? No, it's not. Did I do that? Yes or yes. Andrea. We'll call. Okay, that's a trial run. Trial run. Let's try. Let's try this again. No, it's supposed to be yes or no. All right, hold on a sec, guys. Let's try that again. Here we go. Here's a link. Here's a different link. Try that one. Yes or yes. Let's try this one more time. Just click the link and let me know, did you watch the videos from yesterday? And again, I'm not keeping track of who did or didn't. So you don't need to be embarrassed if you didn't watch them. I just want to know. I just want to know what our starting point is here. So let me know. I'm going to try and use this to do some polling every once in a while. To get a little bit more feedback about students, how they're feeling when we're, how much they're done of a specific thing, or how they feel about a particular concept. Do we need to go back and review? This will be useful to figure that out. Okay, so it looks like it's actually a pretty even split. It's like basically half did and half didn't. That's kind of a bummer. There's more no's than yes though. So okay, so here's what we'll do then. If I start going through this process and explaining it, and you haven't watched these videos, I think that it is going to be problematic. I think it's going to affect your understanding. So how long are these? 5 minutes. I'm going to give you an opportunity to actually watch them right now, and then I'm going to come back. So what is this? 5 and 5. So you need about 11 minutes. So 8:48, 8:50. I'll come back at 8:50. Okay. So watch these two videos here. And they're going to give you a little bit of an introduction to DNA and the process of transcription and translation, just in its very basic form. And then we're going to talk about it together. I'm going to go through these concepts together as a class. So, okay, so I'll see you at 8:50 then. Hey, guys. Okay, hopefully at this point you were able to watch those videos. Sadie, I did get your message there and responded to it. Hopefully you are able to clear your browser cache, and I left instructions there in the chat, and then able to get that going. If you're not, please email me, and I will follow up with you for sure, and we'll make sure we get that going. One thing to mention, I just realized as I was sitting here that I got a bunch of student emails yesterday evening. I normally respond to those before I start class in the morning, but my notifications on my phone didn't work. I didn't get a notification for any of these emails. I've got a whole bunch actually. So apologies, I haven't gotten back to you if you emailed me very recently. And the moment we have a break here, I'll get back to you. Okay, so don't worry, those responses are coming. Okay, so you watch those videos. You may have questions. I'm going to discuss some of the concepts in those videos in a little bit more detail now. If you still have questions about the videos that I don't answer, please be sure to put them in the chat. So they mention in the video that DNA, otherwise known as deoxyribonucleic acid, is a molecule, a big long molecule, and it is a instruction manual basically for building proteins. So the key thing here is that it's for making proteins. Obviously, there are other structures in your cells that are other than proteins, but those are built by the products of proteins. So the DNA doesn't directly assemble anything else. It just assembles proteins, and then those proteins potentially go on to build anything else that needs to be built that isn't a protein. So it's a protein building instruction booklet. Oh, okay. Okay. Sorry about that, Sadie. And thank you for doing that, Bree. If it's not like resolved by the end of the day, let's investigate that, because that's a huge pain. There's been actually quite a few problems with Brightspace this quad, which is a little bit concerning. Anyway, we pay a lot of money for it. Okay, where was I here? Right, proteins. So what do proteins do? All kinds of things. So proteins, like I mentioned, build other things. For example, like, where do you get a Golgi from? Well, a Golgi, it does have proteins in it, but it isn't entirely protein. It has plasma membranes and sugar complexes in it and other things. And those are just all assembled by proteins. So proteins can build things themselves. They are used to maintain all the various biochemical functions of the cell. They're used to make, obviously, all parts for cells, so they can be used to replace tissues. So all of the functional stuff, basically, within your cells is mostly made of proteins. So that would be the structural role of protein. It's the stuff in the cell is made of proteins primarily. Okay, so that's the structural component. Proteins also act as catalysts, which you may have discussed in chemistry. Hopefully, actually, maybe not. You're not all taking grade 11 chemistry, but if you are, you've probably come across the idea of catalysts. A catalyst is a chemical, a molecule, a substrate that changes the rate of a particular chemical reaction. And so enzymes do that. They allow chemical reactions to take place in the cell that otherwise would not occur by themselves. So enzymes are operating all over your body, doing all kinds of things. So we're going to get into some detail in the next unit, animal systems, discussing digestive enzymes, which are the enzymes that break apart all the nutrients that you eat into their components so that you can absorb them and then use them in your body. So that's one function. Enzymes can take big molecules apart. They can also build up molecules, or they can force chemical reactions, synthesis reactions, to make copies of molecules and things like that. So we're going to talk about some enzymes that do that as well. So enzymes are basically the little chemists of your cells. They are zipping around doing chemical reactions, allowing the biochemistry to take place within your cells, and they're made out of proteins. So obviously that's pretty important functionally. So the last function, or the other function, the third function, is that they are used as communication molecules. And those communication molecules are known as hormones. Why shouldn't you highlight in the same color? Hormones. So if you want to send a signal from one cell to the next, let's say that your carotid artery wants to let your kidneys know there is too much water in the circulatory system. And this is real. So if you have too much blood volume, that is, you have too much water in your body, your pressure in your carotid is going to be too high. And it's not just the carotid where there's baroreceptors. Baro is pressure receptors, but there's other locations for pressure receptors as well. And so they're going to release a hormone. Love, are you behind me here?
Yeah, sorry. I'm just
What hormone does your circulatory system release that tells... What is it?
I said, oh God. Okay, keep going.
That tells your kidneys to filter out water.
Filter out water?
Yeah, is it angiotensin?
Like it's the renin-angiotensin system?
Yeah, which is the hormone that says too much blood, too much water.
Yeah, it's the angiotensin system.
Angiotensin. Okay. So again, protein,
Renin.
hormone. It's what? And renin is the other one.
Renin is the other one.
It's a cycle.
Yeah.
Okay. Okay. So there's often multiple hormones that are involved in cell signaling, but those or tell the kidneys, hey, too much pressure, too much water here, and then that will turn on some functions in the kidneys that are going to remove some water from the body. So in other words, you need to send a signal at a distance. There are some cells that are far away in the body that need to tell some other cells to do a job. When you go through puberty, your pituitary gland in your brain becomes active, and it sends out follicle stimulating hormone, FSH. You may have learned this in health class. I'm not sure if they still talk about this stuff in health class. They used to. Which is a hormone, again, just a protein. And then that is what turns on your production of secondary sexual characteristics. So, for example, it activates production of testosterone or estrogen or progesterone, which in turn is going to cause body hair growth, secondary release of growth hormone, and which is going to cause all of this secondary sexual characteristic development that you see in humans. So that's all hormone signaling that's happening. So there's tons of different hormones. Insulin is a hormone, et cetera, et cetera. So there's proteins. Basically, what I'm trying to get at here is that proteins are the functional things that do all the things. Okay, they're the chemicals that do all the different functions of the cells. They're the structures of the cells. They're the signaling molecules of the cells. They do all the chemical reactions in the cells, and so they are the stuff that you need to make in order to get your cells operating. And the DNA is an instruction manual for building proteins. So, and if you just watch the videos, they discuss how that works very briefly. And we're going to talk about that system in more detail. So, a little bit more about DNA before we get into transcription and translation, which is the building of the proteins. That's obviously your hereditary material. So, because it makes all the things, that is what makes you unique. Other than your experiences and your environmental influences, which also contribute to your uniqueness. But what you get from your parents is the genetic material. It's what makes twins identical twins anyway. They have the same DNA, which basically means that they have the same instruction manual for building cells. So if you have the same cell building instruction manual and all your cells have it, yeah, you build things pretty much more or less the same. Environment makes a difference. And so, especially if identical twins are not raised together, they don't look as similar. But they're still identical twins. It's minor. It's minor. So the environmental influence, especially on looks, appearance, is fairly minor. But there is an environmental influence as well. And where do you find that? Well, you guys already know this. This is in the nucleus. This is where you keep your DNA. And almost all cells have that. So obviously, if the DNA is making all the things, all the functional stuff, they're the instructions, you need that in almost every cell because you got to be building and replacing things that are breaking down and doing your cell functions. And in order to do that, you have to have the instruction manual in every cell. Mostly. So there are a few cells that don't live very long, 14 days or 20 days. A good example of that would be red blood cells. In which case, having a nucleus in a red blood cell is actually counterproductive. It takes up a bunch of space, and red blood cells don't require the services of a nucleus for anything. They're just transport tubes, basically, transport inner tubes where you pop some oxygen on it and unload the oxygen. They don't really do much else. So in that case, you don't need a nucleus, and they don't have a nucleus. So we've evolved red blood cells that don't have nuclei because they're unnecessary, which just makes sense. It's more energy efficient to produce transport cells that don't have them. So evolution favored red blood cells that do not have a nucleus. There are a couple other minor places in the body where you find enucleated cells, but the red blood cells is probably the one you're going to come across the most often. Now, that being said, that means that red blood cells can't make copies of themselves. So they have to be produced in via other cells that are going to make the red blood cells. They're found in your bone marrow. And it's kind of an interesting process because it doesn't work like mitosis because obviously they're enucleated. So there's like an extra process that occurs there to make them enucleated. So anyway, we're not going to get into detail on that in this course, but you can investigate that on your own, or you can take a whole course about that in university. But you have pieces of DNA inside of your cells. Keep in mind that we're going to keep this human-centric. So when I say there are 46 pieces or 46 segments of DNA, I mean in humans. You are going to do a project in this unit where you pick a species that has between, 6 and 8 pieces of DNA. And those pieces of DNA are called chromosomes. I'm sure you've heard of this before. So when you talked about mitosis last year, you would have talked about this idea that you're replicating chromosomes. Chromosomes are just the pieces of DNA. Okay? And if you unravel them, you will notice that the DNA is a double helix. That's the name of this twisted ladder structure. It's basically a ladder where you take the top end of the ladder and you twist it one way. And then you take the bottom of the ladder and you twist it the other way. That's a double helix. And the reasoning behind that, why that shape is favored from an evolutionary perspective, is that you can take a huge long molecule, and once you twist it up into its double helix, it takes up a lot less space. So you can condense it and compress it into a much smaller area, which is very useful for cells. I mean, when you're not using specific pieces of DNA, you have to keep them somewhere. So, and they can't take up the entire cytoplasm. So, this system of storage for the information is very efficient. It's also pretty protective because the outside part of the DNA, and we're gonna name all these in a minute, but the backbone part, the part on the outside, I shouldn't use pink. That's really hard to see. Use green maybe. This part, the backbone part, doesn't actually contain any information. It's identical all the way down the backbone. And so what you're doing is you're putting on the outside, the part that's exposed to chemical damage, radiation, etc. You're putting it on the very outside of the molecule, which has a protective effect. You're basically preserving the delicate code, which are the rungs of the ladder, the middle part, and you're just keeping it on the inside. So you can see how this structure could be favored from an evolutionary perspective. It's pretty stable as a molecule. It doesn't readily chemically react at least not under cell conditions. So it's a pretty good storage molecule for information. As I mentioned, the sides of the ladder are comprised of alternating sugars and phosphate molecules. Okay, I've drawn just, whoopsie daisy. That's not what I was going for. Sugar and phosphate molecules. Sugars, you may or may not be familiar with their shape. They form a pentagon, basically, where all of the points of the pentagon are carbons. And the actual shapes of these molecules is not super important for grade 11 biology. I'm not really going to ever ask you that. But just be aware that if you take grade 12 bio, you spend quite a bit of time analyzing the actual chemical structure of these, because it's kind of important to understanding its chemistry. But we don't really need to know that for this course. But anyway, that's a sugar. So when you see a little pentagon-shaped molecule, you know that that's a sugar. And a phosphate. Okay, so it goes a sugar stuck to a phosphate, stuck to a sugar, stuck to a phosphate. Hopefully you remember phosphate from grade 10 chemistry. It is one of the polyatomic ions. It's PO4 3-. And phosphate is extremely important biologically. You will see phosphate come up again and again and again because a lot of biological systems use phosphate as part of their functional unit. So later on, in the animal systems unit, we're going to talk about, ooh, what just happened there? Whoops. Sorry. We're going to talk about a molecule called ATP, which is extremely important. It's the energy molecule of your cell. And the P in that is phosphate. It's adenosine triphosphate. There's 3 phosphates attached to an ATP. Anyway, phosphate, very biologically important. So, getting back to the ladder. Well, what are the actual rungs on the ladder? So, if I were to draw out this ladder, okay, we got our 2 sides of the ladder. And then we have 4 types of rungs that exist. And you know what? I'm going to try and color code these. Why don't I color code them? I'm going to color code these ones red and orange for adenosine and thymine. And I'm going to take guanine and cytosine and make them 2 different shades of green. And I'm going to do that to make it obvious that they pair up with each other. Okay, so we've got adenine. Adenine always pairs with thymine. And guanine always pairs together with cytosine. So these are actually the rungs on the ladder. So if I were to go through and label these, this is adenine. What did I do? Light green. Cytosine. Cytosine. Guanine. Adenine. Cytosine. And then the one on the opposite side, which is called the complementary base pair, is always the same complementary one. So adenine always pairs together with a thymine. Cytosine always pairs together with a guanine. It's meant to be a G, by the way. Geez, terrible G. Okay, so these are nitrogenous bases. Okay, that's what they're called. That's what the chemical is called. It's called a nitrogenous base. And they make up the rungs of the ladder: adenine, thymine, guanine, and cytosine. Those 4 are really important biologically. You will need to know the names of those for this course and every biology course you ever take. So this is one of those like basic biology facts that you must know as a biologist. Is that the 4 base pairs of DNA are adenine, thymine, guanine, and cytosine. And you need to know which ones pair up with which. So A, adenine, always pairs with thymine. And C, cytosine, always pairs together with guanine. So if it's an adenine on one side, it's going to be a thymine on the other. And if it's a cytosine on one side, it's going to be a guanine on the other. And if it's a guanine, it's going to be a cytosine. If it's an adenine, it's going to be a thymine. And then you can go along and make a code. And that's your code. That's your genetic code. So when we're talking about a genetic code, we're talking about the sequence of letters of these nitrogenous bases as you read along the DNA. And there is a directionality to it. So one side of the DNA is going to be the side that has the code on it. Okay? So I'm going to arbitrarily pick it here. The side that actually has the code is known as the coding. Whoops, can I make that smaller? I wonder if I can. Yes, I can. It's called the coding strand. Okay? And the other side of the DNA, try and pick a color that I haven't used here. Pink. The other side is called the complementary strand. Template. Hold on. I better use correct terminology here. This is called the template strand. So when I say it's the coding strand, what that means is that's the side that you read as the code. Okay? So, for example, if someone was like, "Oh, hey, what's the code of that strand?" Well, you just read it. So in this case, you'd read along, and it would be A, C, G, A, C. A, C, G, A, C. That would be the code of the coding strand. The template strand is not the code itself, but it's a tool to make a new coding strand. It also protects the coding strand because it keeps a opposite base pair across from the code at all times. So just in case you accidentally go by, let's say this DNA is hanging out in your cell and it gets hit by some ultraviolet radiation. Okay, which by the way happens all the time. You're out there and your skin cell gets some UV and it blasts away. Oops. And it blasts away. Oh geez. It blasts away one of your base pairs. Okay? It blasted away one of the nitrogenous bases, and this happens. Well, you have an A left over on the other side. So there are enzymes that come by and look for these gaps, and they say, okay, well, we got an A on the other side. The thing that must fit on this side is a T. So it'll come by and just add a T in on the other side. And so in that way, it gives you a little bit of error protection. If you get an error, there is going to be an enzyme coming along and fixing the error. And that works pretty well for minor damage, which your cells get all the time. If you can just imagine, when you go outside, your skin is being bombarded by ultraviolet light from the sun. There's quite a lot. And so these repair mechanisms are operating in the background all the time. Now, if you get a lot of UV damage and you blast a whole bunch of DNA, chances are you're probably going to cause cell death. That's called a sunburn. So a sunburn is when you are starting to get cells dying because they have so much DNA damage that the cell's like, all right, can't fix this. Apoptosis. Pack up all our stuff. We got to kill this cell. And you can get that in large groupings depending on how bad your burn is. That's when it's unrecoverable. And that's why that's why we wear sunblock. Not just to protect from sunburns, but to also protect from the DNA damage that doesn't cause a sunburn. So where there is just minor damage accumulating, you don't want that minor damage all the time either, because it leads to errors eventually. If you get enough damage, you make mistakes when you repair. Sometimes the mistakes are unrepairable, and you may end up with a mutation in a tumor suppression gene. And if you get enough of those, enough tumor suppression gene mutations, those are called mutations when you alter the code, you are probably going to get a precancerous lesion, or possibly a cancerous lesion if you have enough mutation accumulating. And that damage is cumulative, right? Because you use those same cells for your whole life. It's those cells and the daughters of those cells and the daughters of those cells. You're not getting like fresh, perfect cells from anywhere. It's just the cells you got. So, and then again, that's why you wear sunblock. So, fairly obvious. So, anyway, that's the basic premise of how DNA is put together. So you got a coding side. That's your actual code that you go along and read. And then you got the template on the other side, which is sort of like the protective end. And the template also serves another important function, which we're going to talk about shortly. And that is, if you want to build a copy of the code, you build it on the template. The template is going to tell you how to build a new code, build a copy of the code. And you need to copy the code all the time. When you do mitosis, you need to make a brand new copy of all the DNA. When you want to read the DNA to make something out of it, you got to make a copy. So the template strand is getting used all the time to make a copy of the code. Okay, a little bit of terminology stuff. And by the way, this should 100% go on your terminology list. A gene. We always talk about genes and genetics. The word gene itself, Gene, is a section of DNA, a piece of that code that makes one protein. So when we're talking about a gene, we could say like the gene for hair color, the gene for insulin, the gene for whatever a gene is for, the gene for producing testosterone, the gene for creating neurons. I mean, it's more than one gene, but you get what I mean. Each gene makes one specific thing, one protein. Okay? So when we're talking about genetics, we're talking about the whole complement of genes, all the different sections that make all the different proteins. That's genetics. Or if you want to talk about one specific organism's full complement of genes, we call that the genome, the genome. So a genome is all the different genes for a particular organism. So the human genome would be all of the different genes that humans have. Okay? But it's all based on the idea of the word gene, and gene is a section of DNA that codes for one protein. Okay, and I'm going to use the word gene all the time so that's kind of an important piece of terminology. The human genome, and I just went over what genome is, that is the full complement of all of the different genes in a human, has about 3 billion base pairs. That's billion with a B. So there's a heck of a lot of base pairs. So when we're talking about base pairs, one base pair is just one of these letters. Okay, so like one adenine, that's a base pair. Okay? It's called a pair because it has a pair with it on the other side, right? It has a thymine attached to it on the other side. So that collectively together, that's a base pair. So you got about 3 billion base pairs in your DNA. You can look that up. You can download a text file of, I mean, this is like freely available on the internet, but you can download a text file of an entire human genome. The genome that you can get online it's from a specific person, but it is from the person that they used for the Human Genome Project. Really interesting. Who is that person? I don't know if that's known. Who is the person that we genotyped and coded out their entire genome? I don't know. I wonder if it's known. I wonder if that person is like anonymous or if we actually know whose genome that is. Now, keep in mind that other humans, their genome is going to be very, very similar to that person's genome. All the same genes will likely be there unless that person has some type of genetic illness. So all the same genes will be there, but the exact code will have variations in it from person to person. It's not exactly the same, obviously. Your DNA is not exactly the same as your neighbor or even your brother or sister unless they're your identical twin. So that human genome that you can look up is not exactly letter for letter the same as yours, but it is very, very close. There's only minor differences between humans. So anyway, if you go through there and you look at all the different genes that are present, and we can figure out what is a gene, and based on they have a certain code that says this is the beginning of a gene, and they have a certain code that says this is the end of a gene. So if you read through it, you'll find a spot where it's like, okay, this is the beginning. All right, then you start counting 1, and then you get to the thing that says this is the end. Okay, then you stop and you say, okay, that's one gene. And then you keep reading along the DNA and you look for another thing that says this is the beginning of a gene, and then you find another spot that says this is the end. Okay, now there's 2. That's 2 genes. And so you can do this with a computer. Run through the whole code, and you can just tell you how many genes are present. About 30,000. I'm oversimplifying this a touch. So that's why it's about 30,000. There are some modifying factors here that we're not going to talk about in this course that mean that there's probably more than 30,000, but not a lot more. When I say that, I don't know how much, I never know how much to talk about this stuff that's way outside the purview of the course, but when you're reading DNA, there's also sections that are folded over, and sections that are sort of modifying how that DNA is being read. You don't need to know any of this for this course, but that modifies that actual number by a little bit. So it's not exactly 30,000, but it's close. And as a comparison, if you were to look at a roundworm, an annelid, most of them have around 18,000 genes. Henrietta Lacks, she's the origin of the most famous cell line that is used for research. Those cells are used for virtually every experiment that you do in the lab on human cells. And I worked with Lacks cells. I mean, every cellular biologist has worked with them before. This is a really cool story, by the way, and this is Andrea's question is asking here, is the genome built on Henrietta Lacks cells? And I don't know if that's true. My guess is that it's actually not that, because those cells are taken from Henrietta Lacks's cancer. They're tumor cells, and they have a very special property in that they are immortal. Not each cell, but you can reproduce those cells infinitely. Normally, when you take a sample out of a human, let's say that you take a sample of your muscle cells, okay, and you grow them in a petri dish. You can get them to divide in the dish, and those cells will make new cells, and the new cells will make new cells, and the new cells will make new cells, and on and on and on. But it gets to a point where at some point the cells won't make new cells anymore. They hit like their limit in the number of times they can replicate. And there are a number of factors that are involved there, but there's also the end of the DNA. There's a section on the end of the DNA that gets shorter every time you make a copy. And it has to do with the biochemistry of how you actually make copies of the DNA. So, Henrietta Lacks' cells from her tumors don't do that. They can perpetually be replicated forever. And so somebody figured that out, obviously, and they made batches and batches and batches of this person's cells, which we use for research all over the world. So they are a really cool cell line that you can purchase to use. There's many companies that grow them, and they are immortal cell lines. Really, really handy. They are human cells, and so they have a lot of the properties of human cells, but they also are tumor cells. So it's not a perfect corollary. I have a strong suspicion that that's not the cell line that was used for the Human Genome Project, but I could be wrong. Did you actually look that up, Andrea, or is that a guess? It's not a bad guess, because certainly if you're looking for human genetic material to genotype, you have an infinite sample size there for that. But I strongly suspect that that's not what it is. Okay, she's going to look into it. Awesome. I appreciate it. I will say that there is a huge controversy around Henrietta Lacks' cells being used for research, and that is that they were essentially stolen from her as she died from her cancer, and her family has really not been compensated for the fact that her cells were stolen by some researcher, and then they made a huge profit off of it, and her family essentially did not receive anything. I don't know if that situation has changed, but initially they didn't compensate the family at all, which seems ridiculous. They basically stole this from the family and used it for. I shouldn't be laughing. They used it for research purposes. It's not a pleasant story. Because they made billions. This cell line is extremely valuable. Interesting. I mean, it wouldn't be surprising if they also sequenced her genome. 2013, though, is after the Human Genome Project was complete, though, so that would be after. 2 males and 2 females. Okay, cool. So they had a relatively small sample size there, but no specifics on who they are. I wonder if they're anonymous. Interesting. Because we use that code from the Human Genome Project for all kinds of things. Now, I will say that when you're doing comparative studies and trying to figure out what gene does what, at this point now, we have the complete genome of probably hundreds of thousands of people that we use for that research. Maybe more than hundreds of thousands. Maybe it's possible that it's millions at this point. So once we figured out how to do it, that process for actually sequencing an entire genome is extremely fast. It's much, much faster than it used to be. In the old days, like when they did the Human Genome Project, it took them like 5 years to genotype one person. And they had to do it, oh, it was so laborious. Just imagine, there's like 100 labs participating, and collectively, it took them 5 years to sequence one person. Now with shotgun sequencing, which is done with computers, where you just do it in tiny chunks at a time, and then have a computer stitch all of the pieces together at the end to make the complete genome, you can do it in like days. So it obviously is way less labor intensive. It's almost completely automated now. So anyway, there's been huge advances in genotyping, but it's interesting to see how far we've come, even in the past 10 years. I mean, we've gone from it taking years and years to genotype one organism to, hey, put a sample of DNA in this thing and send it in the mail, and we'll genotype the entire thing for $30. And then, I mean, it's not that cheap. I think it's $2,000 or $3,000 the cost to get a complete genotype done. But it's really extremely inexpensive. It costs, tens, hundreds of millions to do it the first time, but now you can do it for a couple $1,000s. So if you're ever sending your DNA into something like 23andMe or something like that, keep in mind that they're not genotyping your entire genome. So that they're not going through and figuring out the entire sequence from beginning to end. They actually, what they do is they look at short repeating sequences of DNA that are characteristic of certain things. It is very similar to how they do a genotyping for forensic purposes, like in a courtroom. Except the ones that 23andMe do and stuff like that is a little bit more elaborate than that. So they look at, I'd have to look it up, maybe a couple thousand sequences of DNA. And they look for things like genes that are potentially causing illness and things like that. There's actually various types of services you can get them to do for you. But it's not a complete genotype from end to end. That's considerably more expensive. So, but you can get that done as well. You can be completely genotyped. I don't know what the cost is on that. I think it's several $1,000s. But anyway, researchers do it all the time. That's how we figure out the levels of relatedness. I mentioned this in another lesson when we talked about building phylogenetic trees. We would maybe genotype 7 different species of frog or whatever. So we do stuff like that all the time. It is expensive. We know the functions of those genes for about 50%. So that number is actually from maybe about 5 years ago when I looked that up. So I would guess that it's probably significantly higher now. It's probably 60%, maybe more, in terms of the known functions. When we say that we know the function, we don't necessarily know exactly what the protein is that's made by it. We just know that when you see this protein, this is what the organism typically does. In other words, oh, hey, when this gene is being transcribed a lot, when we use this gene a lot, people get diabetes. Or when you don't use this gene very often, people tend to have schizophrenia or whatever. Okay? You choose. So we know what the effect is of the gene being upregulated, like turned up, used more, or downregulated, used less. But we don't necessarily know exactly what the gene makes, always. So sometimes that's a little bit difficult to figure out. But 50, 60-ish%, we have a general idea of what the gene does. Now, a fairly large percentage of that we do know exactly what it makes. Like we could build that chemical synthetically in the lab often. But we don't have that type of information for a huge percentage of the human genome. So we don't know quite that much. And obviously, if you're going to get into genetics as a profession in academia, part of what you do is that you just figure out what genes do. What happens when somebody has this mutation in their gene? What mutation in the gene causes this illness? That's the kind of stuff that you do. That's your bread and butter as a geneticist. So if you're working with human stuff, anyway, not everybody works in the human area. But anyway, interesting fact. Only about 1.5% of your entire genome actually codes for proteins. You might be thinking, like, what the heck? I thought that was the purpose of DNA. You have all this DNA, and 98.5% of it doesn't actually make proteins? It isn't genes? Yeah. That's weird, eh? So the rest of it, way back in the old days, like back in 2000, that's the old days for genetics, we used to call that junk DNA. And in fact, when I was in high school, this was still a term that was used often, but not any longer. So I crossed that out there. We don't really call it junk DNA anymore. Now we call it non-coding DNA, or the flashy science name for that is an intron. An intron is a part of the DNA that doesn't actually make a protein. It's not the gene part of your DNA. And we used to think it did nothing. But that is a silly guess, in my opinion, because evolution does not preserve things that do nothing. Not very often, anyway. I mean, you do get things ending up hiding away in your genetics, like an appendix or something like that. But typically, over the long term, sections of stuff, really anything, that are pointless and a waste of energy and materials do not get preserved by evolution, because evolution will just say, well, there's a faster way due to random mutation, and then that mutation will be more survivable, will be a better mutation for the environment. And so you just end up not having it generally. It gets selected against by nature. So that's a silly guess, in my opinion. But introns, in fact, are extremely important. So what do? That's those sections that don't have any genes in them are on-off switches. That part of the DNA is what tells you, oh yeah, we need you to make this in this cell. Or, no, this section of DNA, don't read that. You don't read that in this cell. And for prokaryotes, we talked about this previously. I just mentioned it in passing. They don't have that. So prokaryotes don't have introns. Those are bacteria and archaea, because they don't have sections of their DNA that need to be turned off. They are only one cell, and so they just need to make all of the stuff that their cell says that they need to make all the time. But in a multi-celled organism, and actually in a lot of single-celled organisms, because they're fairly complex, you don't want to be building everything all the time. Sometimes you want to build certain proteins, and sometimes you don't. So for example, if you are a pancreas cell, you have a gene for making insulin. Insulin is a hormone that causes your muscles and liver to take up sugar from your blood. If you have diabetes, type 1 diabetes, you do not make insulin. That's what causes diabetes. And you may have heard of people taking insulin shots and things like that if they have diabetes. It's because their pancreas does not produce insulin. So in those pancreas cells, you have to turn the gene on that makes the protein. So that would be switched on. But in your eye cells, or in your skin cells, or in your liver cells, or in your muscle cells, you don't want to make insulin in those cells, because it would probably cause cell death if you accidentally made insulin in those cells. So it's turned off in those cells. So some cells are for making certain things and other cells aren't. And so some genes have to be turned on in some cells, and in other cells it has to be turned off. And that's obviously an extremely complex process. Imagine that how different your eye cells are from your skin cells, or how different your root cells are that make your hair from like your heart muscle cells. Like those cells are so different, but they have the same DNA in them, the same plan. So there's a very complex system of turning things on and turning things off to make all the different types of tissues that you have in your body. And that's what the introns do, the non-coding part. And that's why 98.5% of your DNA is that. Because it's actually far more complicated to control all of the different sections than it is to make the different proteins. Way more information is required to control it. So anyway, that's what the other part does, the intron part. And once you realize that and think about that, it's actually crazy that we used to think that it did nothing, that it was junk. Like, how could you think that? I don't know. I think that's silly. That was a terrible guess on the part of biology as a whole. But I mean, that's science for you. We got to start somewhere. Anyway, this is a little breakdown here of a piece of DNA. I just wanted to show you what the molecules actually look like. So we've got our backbone. So if you look at the backbone, it goes phosphate, sugar, phosphate, sugar, phosphate, sugar. That's the backbone. So it's a phosphate stuck to a sugar, stuck to a phosphate, stuck to a sugar. And if you look on the other side, you will see the exact same thing. So phosphate stuck to sugar, stuck to phosphate, stuck to sugar. Sorry, Bree. A base pair is, let's go back to the middle of the DNA here. If you look at the ladder, the rungs of the ladder, these right here are a base pair. So those chemicals that make up the middle part of the ladder are called nitrogenous bases. That's what they're called. And a base pair is the set of them that makes up the rung. So this set of A and T right here is a base pair. So this is a base pair, this is a base pair, this is a base pair, this is a base pair. And you can go along and read one side of them, and that on the coding side, the one that is the coding strand, and that actually is what makes up your code. So if you go along and just read this A, G, C, that's the code that you read on the coding strand. The other side, the template strand, which would be the opposite side, is not directly the code, but it is responsible in making a copy of the code, and we're going to talk about that process. But it's sort of like a backup plan. It's like having a photograph and then having the negative of the photograph on the other side. So if you ever lose the photograph, you can make another copy using the negative. I just came up with that. I feel like that's a pretty good analogy. Maybe not. Maybe if you've never done photography, it's probably not. But anyway, that's basically how it works. Like, there are 3 base pairs, but 30,000 genes. That's correct. There are 3 billion of these combos. 1, 2, 3, 4, up to 3 billion. Okay? Some sections, remember that a gene is a section of the DNA that makes one protein. So if you go through the DNA and find all of the sections, like, okay, this section makes insulin, this section makes testosterone, this section makes estrogen, this section makes, I don't know, renin. This section makes angiotensin. And you could go through and you can name like every protein. If you go through and look at all the different sections of DNA that make stuff, there are 30,000 sections that make different proteins. So 3 billion letters, but 30,000 words. There's another analogy that's probably helpful. But there are a bunch of other letters in there, 98.5% of the letters, that don't actually make words. They're just like what looks like gobbledygook in between, but they turn the words on and off. Did I answer your question, Bree? I hope I did. I can try that again in a different way. If not. Or if you'd like me to try it again in a different way in the Google Meet, that's also possible. It does. Okay. All right. So a little bit of extra terminology here that I just want to quickly mention. When you hear the word nucleotide, what they mean is a phosphate, a sugar, and the base pair that is attached to it. So when you put all 3 of those together, that's what I have highlighted here in blue, that's called a nucleotide. And DNA, it's said that DNA is built out of nucleotides. So it's built one nucleotide at a time. So all these 3 parts are usually always linked together. And so you can build DNA by building one nucleotide and then the next nucleotide and then the next nucleotide, and that is how you assemble the ladder. So you might have a guanine nucleotide, and then you add on a cytosine nucleotide, and then you add on a thymine nucleotide. So you build them one nucleotide at a time. I told you there was a lot of terminology in this course. It's actually ridiculous. There's so much. I remember taking this in high school and looking at my pile of flashcards and thinking, oh, this is insane. There's so much for me to learn here in terms of vocabulary. I won't pretend I wasn't a little overwhelmed when I was in high school. I was. I sat in your shoes. So I remember doing this exact thing in high school. I had an amazing grade 11 biology teacher who actually is probably the reason why I became a teacher. She's a great teacher. I remember struggling with this in high school with the terminology. There's a lot. And I just wanted to point out this again here, that the phosphates are PO43-. It's exactly the same phosphate that you would see in your chemistry class in grade 10, right? That's the polyatomic ion. And the sugars, I mentioned that the S here, the ribose or deoxyribose sugar, that sugar is called deoxyribose, is a sugar molecule. It's exactly the same sugar that you eat. I mean, the bonds are slightly different. But even though the bonds are slightly different, the composition of it is the same, C6H12O6. It's the same sugar that you would eat, like glucose, like eat at the table, with slightly different bonding. But the composition is the same. It's the same stuff in it. And you'll find that all the time in biology. I mentioned this as part of cell theory, that it's like the same molecules that you see over and over again, but it's just like very small variations of molecules that get used in all these different things. But you just like change the molecule a tiny little bit, and then now it does something else. So I think that's really cool. And it also means that in order to build all this crazy stuff that's in your cells, you really only need to take in a very basic set of nutrients, and you can build all of these different things out of those nutrients. So, for example, where do we get the deoxyribose to make our own DNA when we need to make copies of cells and stuff? Well, you eat sugar. And then you have your cell is able to modify that sugar and turn it into a deoxyribose or turn it into a ribose. So you take something that is just like a basic nutrient that you would eat, and then you make it into like a functional unit that you would use in your cells. So I think that's cool. All right, I'm gonna keep going here. So that's the basics. I forgot about this part right at the bottom of my note here. In terms of organization within your chromosomes, you have 23 pairs of chromosomes, so 46 in total. And when I say pairs, we're going to get into more detail on this in a moment, but you have two versions of each chromosome. One version from your mom, that's your maternal one, and one version of the chromosome from your dad. That's the paternal. Maternal just means from mom, and paternal just means, it means of mom, and paternal means of dad. So that's how you were assembled. You were assembled from half the DNA from your mom and half the DNA from your dad. And so you have a pair of chromosomes for each thing. So chromosome 1 is actually a chromosome 1 from mom and a chromosome 1 from dad. And chromosome 2 is chromosome 2 from mom and chromosome 2 from dad. So there's a pair of each. So you have 46 in total, but you have 23 pairs. We organize them by size. So chromosome 1 is called chromosome 1 because it's the biggest. And if you look at the number of actual genes, proteins that it makes, there's 1,961 genes on chromosome 1. It's pretty big. And if you were to look at chromosome 2, it has slightly less, and on and on. Chromosome 21 actually has the fewest. It has between 2 and 300, again, depending on how you measure. And I'm not going to get into the specifics of that in this course. But 21, what about 22 and 23? Well, by chance, 22, which is actually shorter than 21, like physically shorter, it actually has slightly more genes than 21. So we organize them by size, just by pure length. But 22 is actually a little longer, but it has fewer genes. So, or sorry, the exact opposite of that. 22 is longer. No, wait, 22 is shorter. 22, because they get shorter as you go up. So 22 is shorter, but it actually has more genes than 21. And 23, the last pair, and I use the word pair very loosely here, are your sex chromosomes. And the sex chromosomes have the fewest by far. So, and they're kind of left out of this equation here. And we'll talk about why later. Because the sex chromosomes are what decides which sex you become as an adult in sexually reproducing organisms like humans. So, okay, I'm going to keep going here. That brings us to this idea of the karyotype. So a karyotype is a photograph of your chromosomes. How do you get that photograph? Well, we usually do karyotypes while you are developing as a fetus. Okay? So you are in an amniotic sac, developing as a fetus, and that's usually the point where it's often a good idea to take a look at the chromosomes. We don't usually actually do this that often anymore because there are some risks associated with it. You need to sample the amniotic fluid with a needle, and there's obviously risks associated when you puncture the amniotic sac. And there are other ways to test for this now, which, so for example, I just recently had a child, and during the pregnancy, we did this testing as a maternal blood test. So you can actually do similar testing by sampling the mom's blood. And I'm not going to get into the details of how that works because that sounds, it doesn't initially make sense. It didn't initially make sense to me. But anyway, it's not relevant for this course, but we rarely do this procedure anymore, but this used to be very common. And so what you do is you sample the amniotic fluid. You use a chemical to lock the cells in metaphase. And remember that in metaphase, all of the chromosomes are visible. I don't know if you remember that from grade 10, but when you did mitosis, during metaphase, all the chromosomes become visible. And then you give them you give them the chemical to lock it in metaphase, and then you use a dye to make the parts of the chromosomes, that's the banding patterns on here, become obvious. And the dye sticks to different sequences of DNA. And because it does that, some regions of the DNA will be darker than other regions. Okay, so you've got, and then you end up getting, you can take the DNA and take a tiny little syringe and move the chromosomes around. It's a little complex operation under the microscope. And then you take a photograph of it, an actual picture. And then this used to be done more recently with computers. In the old days, we would do it by hand. But then you got to go through and actually match up the chromosomes. And remember that they come in pairs, right? You have basically the same chromosome from mom and from dad, which is the maternal and paternal chromosome. And because those two chromosomes are largely the same, they have the same genes on them, we call those homologs. Or another way to say that is they are a homologous pair. So we use this terminology actually in the last unit in diversity, because I don't know if you remember this. When we were talking about phylogeny, we talked about how we compare the DNA between species for the parts of the DNA that are the same parts. And for example, we have 98% DNA the same as chimps. Okay? I don't know. I used this terminology back in the last unit, but I don't know if you remember. So that 98% that's the same, we call that 98% homologous. Homologous doesn't mean identical, but it means that the parts do the same thing. They code for the same stuff. Okay, that's what a homolog is. It has the same function. So by the way, you can use the word homologous outside of this context. You can go out in nature and see a butterfly has wings for flying, and a bat has wings for flying. Those structures have the same function, so you would say that those structures are homologous. Oh no, wait, no, that's wrong. They have different origins. No, they have to come from the same origin. Let me try that again. Birds have a wing structure that is evolved from an arm, basically. And bats have a wing structure that is evolved from an arm. They have the same genes, actually, that make those structures, roughly. So those two structures would be homologous. They have the same origin, okay? There you go. That's much better. That's much better. A butterfly has wings, and a bat have wings, but the origin of those wings is completely different. They are completely different structures. They just have the same function. Those are called analogous features. Sorry, I completely mixed that up a second ago. It's called an analogous feature when they don't have the same origin, but they have the same function. And it's called a homologous feature when they have the same origin. So in this case, these two pieces of DNA have the same origin within the species, within our evolution, and they have the same function. So we would say that these two pieces are homologous to each other, same genes. Okay? Sorry for mixing it up there. I do that sometimes. I got it right now though. So we've got a chromosome 1 here with a mom and a dad component, okay, that are homologs. We've got chromosome 2 with a mom and a dad component, maternal, paternal. 3 maternal, paternal, 4 maternal, paternal, 5 maternal, paternal. And you can see that they look the same under the microscope. They're not identical to each other because they will have different genes. Obviously, the genes you got from your mom and your dad are not identical to each other, but they are largely the same, okay? I mean, they're both human, so they're going to be largely the same genes, if not identical. Now, they could be identical. It's possible, but it's unlikely because there's so many different genes that you're probably not going to get two that are exactly the same. Okay, so 6 maternal, paternal, 7 maternal, paternal, 8 maternal, paternal, etc. Okay, going through all the way up to 22 maternal, paternal, and notice that it's the smallest. So they're organized here from largest, where 1 is the largest and 22 is the smallest. Okay, but I remember that I said that there are 23 pairs. The 23rd one is either an X and an X. That would be if the person is female. Or if they're a male, there's going to be one X here. And then this one wouldn't be here. It'd be an X, and then the Y is just a little tiny guy over here. Okay? And that's what determines if you are male or female sex. So if you've got an X and a Y, that is going to initiate the development of male sexual characteristics. And if you are a female, you'll have 2 X's, and that initiates the female sexual characteristics in utero. Basically, in embryonic development, if you don't tell the developing embryo to become male, the default is female. So if you don't add any of the hormone signaling in that says male and you just leave it out, you get female. So it's sort of like female is the default, and if you have the genes, the extra piece here for male stuff, that's going to divert the process to male. But if you're missing this, you're going to end up female because the default is female. And that only matters when we start. We're going to be talking about genetic illnesses a little bit later. And when we do discuss genetic illnesses, you'll see how this comes into play. It's kind of important, actually. that if you're missing, let's say for example you only had one X. Okay. I wonder if I can blank this out. I might be able to. Oop. Can I do this? Oops. I'm struggling, guys. What about this one? Perfect. Okay, let's say that you only had one X and no Y. Okay? So obviously this is a problem. You don't have 46 chromosomes. You're supposed to have 46, but you have 45. That's a problem in itself. But this person, and this is survivable by the way, is going to end up being female. Because they have the signaling for female, but they don't have the second signal for female. But there is nothing here that is going to indicate male change from the default. So this is going to end up being a female that is born under these circumstances. Anyway, that's kind of neither here nor there right now. But the important thing for you to know about this karyotyping here, and this is actually on the quiz too, so it's kind of important, is that we organize pieces of DNA in organisms first by size. So 1 to 23 is done by size from largest to small. 1 to 22. 1 to 22 is done from largest to smallest. Then we look at banding pattern. So banding pattern would be the patterns on here. And you'll notice that some of these look very similar in size. And obviously, yeah, that's problematic, especially if you're organizing them by hand. But so what you do then is you look at the actual banding pattern on these. So you might notice that like there is a band here, and then a band here, and then a band here. And then the other one that has those same bands in the same places, those are going to be the homologs. So we organize them first by size, then by banding pattern, and then lastly, if we have ones where the size and the banding pattern look similar, like these two, which have a band here and a band here, this one has a band here and a band here. These look similar, the similar sizes, similar banding pattern. Lastly, we look at centromere location. So how far down on the chromosome is the centromere? And then when you compare all of those features collectively, then you can use that to organize them. Okay, so those are the three ways that we use to organize them into groups from chromosome 1 all the way down to chromosome 22 in humans. It's a different number depending on the organism. And we also make an exception. We don't organize the sex chromosomes by size. We kind of leave them to the end. So I should probably just add that in here, that sex chromosomes that go at the end. Okay, a little bit more terminology. Okay, I mentioned that there are 23 pairs of chromosomes. That number of pairs that you have is called the haploid number. And I have mentioned this before, but that makes up, if you have the haploid number of chromosomes, you only have 23. And keep in mind that this person above here has actually 46 chromosomes, right? Not 23. But if you only have 23, you would have the haploid number. And it is important that if you're making a sperm or an egg, a gamete, they're called, you need to only have the haploid number of total chromosomes in your cell. That is half of the full complement. But when we say that the haploid number of an organism is basically its number of pairs. Okay, so there are 23 pairs in humans, and so the 23 is the haploid number in humans, or the 1N number. I already mentioned here that we arrange them by size from largest to smallest, but the total number of chromosomes that we have, if you count them all, is 46, and that number is known as the diploid number. Diploid essentially means the full complement of chromosomes, all of them. And all of your adult cells, which are known as somatic cells, terminology. Somatic cells, your somatic cells, your adult cells, have diploid DNA. They have pairs, and there are 46 total chromosomes in them, if you were to count up all the pieces. So all of your adult cells have 46 pieces of DNA, the diploid number. If they only had half the haploid number in them of total chromosomes, your cells would not be able to survive. They can't exist in a haploid state. There is only one type of cell that exists in a haploid state, and that is sperm or egg. And they are designed to operate as haploid. But other than that, cells cannot survive as haploids. The non-sex determining chromosomes, those are the ones that are 1 to 22 in humans, are known as autosomes. So an autosome are the chromosomes that don't determine the sex of a organism. Okay? And if something happens to one of those chromosomes, you have a genetic illness on one of those chromosomes, it's called an autosomal genetic illness because it's not part of the sex chromosomes. And the reason why we differentiate between the two is because the inheritance patterns are different. How you figure out whether or not you can pass it on to your kids is different depending on whether it's on an autosome, non-sex determining, or an allosome. An allosome is a sex determining chromosome. Sometimes they're just called sex chromosomes. Sex chromosomes is fine, by the way. You can absolutely call them that. But allosome is like the official scientific name for them. So we've got autosomes, which don't determine sex, and allosomes, which do determine sex. So I mentioned that the above karyotype that we were just looking at together there is a female because there are 2 X chromosomes in the karyotype. There's a question on the quiz about this as well. But if this was male, they would end up having this for their allosomes. That is an X and a Y. Okay, so there's 2 X chromosomes in the above example because it's female, and then for a male, there'd be an X and a Y. A lot of information there about DNA. If I'm ever going too quickly, don't be afraid to let me know in the chat. I am, with no feedback, the way we're doing things right now, I do have a tendency to go a little bit too fast, and sometimes I don't notice. So what we're going to do now is we're going to go through the process of how you go from a piece of DNA to a protein. Okay? That process is called protein synthesis. Synthesis means to build. Or it's called transcription and translation. Transcription and translation are the 2 parts of protein synthesis. Okay? This process is often called the central dogma of biology. And by that, it means it is the most important concept in all of biology. And that's really not an overstatement. Once we learned this, how this works, I mean, this is really where we really started to learn about genetics. But once we learned how this process worked, this opened the door to pretty much everything that we understand about life at the biochemical level. How do we know things are related? How do we figure out what does what inside the cell? How do we figure out why one person gets a genetic illness and another person doesn't? How do we use bacteria to hijack this process and build stuff for us, which we do all the time? How do we fix genetic illnesses? How do we predict if people are going to get genetic illnesses? So again, choose an area of biology. Understanding this concept is pretty much integral to understanding and developing knowledge in every single area of biology. And I can say that unreservedly. Ecology, biochem, molecular biology, physiology, medicine. You honestly, you name it. Understanding this process is at the core of everything. And that's why it's often called the central dogma. It is the most important thing to understand about biology. So I don't want to oversell it, but this is one of those processes that is in the background of every single sub category of biology. So if you're interested in biology, if biology is a thing that you are going to make part of your repertoire, this is the one process that is applicable to everything. It is probably the most important thing that you ever learn about biology. So again, I'm not trying to oversell it here, but you're going to want to make an effort to get a fairly working understanding of how this process works. And we're going to talk about it fairly generally in this course because you do need to understand this process. But we're not going to get into the biochemistry real deep of this, like what enzyme does what and what's the order of things. And you can really dig into this, into the chemistry component of this, and you do in the grade 12 unit. So if in grade 12, there is an entire unit that you do just on this one process. Because it's extremely important. I'm just going to obviously give like a more cursory explanation of it here. But just be aware that this is like one of the core concepts of biology. It's really important. So if this is something that is really nagging at you and you're not really understanding what's going on, please come and talk to me during the Google Meet, because I would love to go over this process again with you. I will use some different analogies. I will make sure that you're comfortable with this concept. This is one of those ones where you're not going to be like, I didn't really understand protein synthesis, but that's okay. This is one of the ones where you're not going to want to walk away until this makes at least some degree of sense to you. All right. So that being said, let's get back to DNA. Okay. So here we are. We have a strand of DNA. Okay. And I mentioned that DNA has two sides. It has a coding strand and a template strand. Okay? The coding strand is the code. And you might be wondering, well, how do you know which one is the code and which one is the template? They basically look exactly the same. So there is a way to figure out which is which based on... Oh, where's my pen? If you look at the ends of the DNA over here, you look at what is bonded to the sugar on the end here, and it'll have a bond on here with a particular OH group or whatever in a specific location. Okay? And then if you look on the other side of it, it'll have that OH group in a different position. Okay? And by looking at those, you can figure out which side is which of the DNA. And then the top piece here at the top will have the opposite on it. And you only read it in one direction from one end to the other. But I'll be very clear here. In the grade 11 course, you do not need to know that. So let's not worry about that at all for this course. And I'm going to tell you which piece is which. Okay? The bottom piece here is the code, and the top piece here is the template. It's not always drawn this way, where the bottom is the code and the top is the template. Sometimes it's switched, where the code is on top and the template's on the bottom, which is very confusing. I wish they would just pick a standard way of drawing it or writing it, but that doesn't exist. So I'm just going to tell you. And to keep things consistent, I am always going to write the code on the bottom and the template on the top. That is just me. I'm going to keep it consistent for you because it's confusing otherwise. In grade 12, you're going to talk about the chemistry of how to identify which one is which. And then you will determine that, because you will understand how to pick which one is the template and which one is the code. But you do not need to know that for this course, and we're not going to discuss it. So I'm going to tell you, the bottom is the code, okay? And by that, I mean, if you were to read along the bottom strand of the DNA and read all the base pairs, that is how you get the code. So for example, the code of this particular strand is ATGCGGTACTTA. Okay, all I'm doing is I'm just reading along the code. Okay, I'm going along and just reading it. The top strand here is the template strand. It is not the code, but it is essential in making copies of the code. That's what the template is for. It is a template for making copies. And you maybe in art have used templates before, or maybe even in software you've used a template, which is basically like a tool that helps you make a copy of something. That's what it is. So we've got a code, and we've got a template, which is a tool for making a copy of the code. And these are normally stuck together. Okay, you already know that. These are bound together. They're drawn here slightly apart because I'm gonna use that property to help. But these are normally directly touching each other. Okay, they're bound together. So what we want to do is we want to make a copy of the code. I want to make a copy of this, and I want to take it out of the nucleus, which is where it is now, and I want to bring it to something that can build a protein for me. Now, we just discussed this, but and by discuss it, I mean you guys review this on your own, but what would be that thing that I would take it to that is going to build me a protein? Anybody remember? Throw it in the chat if you can remember. What's the thing that does the building? You got it, Mila. Quick on the draw. It is, in fact, the ribosomes. So the ribosomes are going to do the building, but they're not in the nucleus. And we don't want to keep our DNA outside of the nucleus because it's dangerous out there. There are all kinds of enzymes zipping around. There are reactive oxidative species that could potentially damage the DNA. So we're going to keep the DNA locked up tight in the nucleus where it's safe. And instead of taking it out and feeding it into the ribosome directly, which is dangerous, we're going to make a copy of a piece of the DNA, one gene at a time. So we're only going to use one gene's worth of DNA. And then we're going to send that out to the ribosome. And then the ribosome is going to read that and make a protein. That's what it does. Okay? So that there are two parts to this process. The first part is making a copy of part of the DNA. That is called transcription. Okay? Transcription means to make a copy. That is what the word transcribe means. If you were a medieval monk, your job, a large part of what your job is to be a scribe or to transcribe books. Believe it or not, that's what most monks did in the Middle Ages as their primary function, is that they read books and they made copies of those books. That's how books were made before the printing press was invented, which completely changed the way that information was distributed after that. But and in the West anyway, it was invented quite a bit earlier in the Far East. But anyway, transcribing something is simply just to make a copy of it. Okay? So the first part of this process is to make a copy, transcription. Then you're going to take that copy and you're going to turn it in to a protein. That is, you're going to take it from one form and convert it into another form. And we have a word for that in English as well, and that is the word translation. Translation means to convert from one form into another form. And it's often used in language to mean to convert from one language to another language, but that translation actually has a much more broad meaning than that. It actually just means to convert from one form to another. But we use it in language to mean language translation. But there's all different types of translations that exist. Maybe in calculus or in physics, you've talked about physics translations, where you're talking about simply moving an object from one position to another. That's translational motion. That is also that means it's the same meaning of the word, which is to convert from one form to another. And that in that case, you're converting your position from one form to another, but that's translation. So translation just means to convert from one form to another. So again, two parts. One, you make a copy, and two, you convert it from that copy into a different form, which would be a protein at the end. Okay. So sometimes this process is called transcription and translation because those are the two things that you're doing. You're making a copy and you're converting that copy into something else. All right, that's in basic what you're doing. And if you watch those two videos, you would have seen that that is what's happening in those videos when they're talking about actually making something out of the DNA, transcribing it and then translating it. For some reason, because these words both begin with T, they are very often confused by students. I often see students calling the first part here translation and the second part transcription. There seems to be a lot of confusion around these words. But in order to prevent that confusion, really think about what the word means. So to transcribe something means to make a copy. That's what a transcription is of anything. If you see a note somewhere and then you write that note down against somewhere else, you've transcribed that note. You've made a copy of it. And translating it simply means to convert it to a different form. So once you have like an understanding of what the actual meaning of those words is, it's pretty hard to mix them up here because the processes are completely different from each other. So I would try and come at it from that direction and actually like understand what the words mean. And then that's going to help you a lot with naming each of these stages appropriately. So I just realized that I'm about to run out of time. When we come back at 11:05, we're going to get into the actual process here of transcription and translation. How do you go from having a piece of DNA to having a protein, which is a completely different thing? Okay, and so I'm going to let you guys, I'm going to pause right here. If you haven't had a chance to watch those videos, I know some people are having some difficulty, take an opportunity to watch those two videos. They are a great overview of what we're going to discuss, and it is a little bit complicated. So enjoy your lunch or whatever you eat at this time, snack. And I will see you at 11:05. Hello, guys. Let me know in the chat if you're here for the second half. Whoops. Ooh. All right. Let's talk about transcription and translation. So, just imagine here, a lot of the stuff that I'm talking about here, the basics, the sort of a summary of what I'm talking about is over here on the right-hand side. But I'm gonna add a fair amount of detail to that. So, again, we have a coding strand and a template strand, and together that makes our double-sided DNA. What we want is to make a copy of the code, that is transcription, transcribe the code, so that we can take that code outside of the nucleus and build a protein, that's the translation part, with a ribosome. Okay, the ribosome is going to read the message that we make. So, an enzyme is going to come along and read, well, not read, sorry. An enzyme is going to come along and split the DNA up first. So it's a ladder. Like I said, these two strands are going to be attached to each other, and so this little protein is going to come along and whoop, zip along and unzip it. Okay, so that's the start. It gets unzipped. And then another protein is going to come along, another enzyme, lots of enzymes involved here. You don't need to know the names. not in this course anyway. Another protein is going to come along, and what it does is it goes to the template, okay? Not the code, but it goes to the template, and it looks at each base pair, and it grabs the complementary base pair. So thymine, the complementary one would be adenine. Adenine, the complementary would be thymine. Cytosine would be guanine. Guanine would be cytosine. It's going to come along and find the complementary base pair and attach it to each one as it goes across. Okay, so I'm going to do these ones in green so you can see them. So this enzyme is going to run across and go, okay, this is a T. I've got to attach an A. This is an A. I've got to attach, normally it would be a T, right? T goes with A. But there's one exception here, and that the strand that we're building on here is a slightly different chemical composition than DNA. The sugar in it, and remember going back to our page up here, there are these sugars, right, as part of the backbone. These are the S's here, the sugars. Instead of the sugar being deoxyribose, the sugar in this molecule that we're going to build is just called ribose. Okay? So instead of it being DNA that we built, if you remove the D part, the deoxy, you just get ribose. And so then it becomes ribonucleic acid. Ribonucleic acid is RNA. Okay, so what we're actually building on here in green is the RNA. Okay, this is an RNA sequence that we're building. And specifically, because this RNA is going to be a message from the DNA to the ribosome, we add a little tiny M at the beginning here, and the M stands for messenger. So this is messenger RNA that we're going to build. Okay, mRNA. mRNA does not have thymine. That's the T part. So when you're building an mRNA, anytime that you would put thymine, you have to use an alternative that mRNA has instead of thymine. This is a little bit confusing, and it's called uracil. Okay, and it's shown with a U. So it's going to go AU because uracil instead of thymine. Then we have a C. C pairs with G. G pairs with C. C pairs with G. Oh, I better make my G's and C's really obvious here. Sorry. G. C pairs with G. A pairs with, normally it would be T, but there is no T's in mRNA, so it's going to be U. T pairs with A. G pairs with C. A pairs with, normally it would be T, but now it's a U. A pairs with, normally it's T, but it's U in RNA. And T pairs with A. Okay, so it's gone along, and this is why the complementarity here is so valuable, or one of the reasons it's so valuable, because it allows us to use the bonding there, the fact that it only bonds with one particular chemical, to essentially make a copy. Because if we take that mRNA that we've just built and we lay it down over top of the DNA, you'll notice it is identical. It is exactly the same as the original code. The only difference is that the T's are switched with U's. Okay? But other than that, this looks exactly like the original code. So, and that's the purpose of having the template. Remember, the word template is just a tool used to make a copy. Okay? So the template strand is used to build that copy. So we have this mRNA, and that process of making that strip is called transcription. And you'll see it below here. This is it right here. It's just a copy. Okay? And if we look at this copy that we've just made, you'll notice, first of all, that it starts with these three letters. AUG. AUG in mRNA is known as the start codon. Anytime those enzymes are running along and they see AUG. That means that's the beginning of a protein. Okay, AUG is known as the start codon. So if you look at it, I wrote them above here. If you're looking in DNA, remember that the U's are actually T's. So a start codon in DNA, which I've highlighted at the top right here, a start codon in DNA is ATG. So going back a little bit, you may remember that I mentioned that we looked through the human genome and figured out that we have about 30,000 genes. So how we do that is we go through and look for starts. So every time we find an ATG, that's the beginning of a new protein. Now there's also stop codons, which mean the end of a gene. We'll look at those in a minute. But those are found at the end of every gene, and then that way we know that that's the end of a particular gene. Okay? So, but ATG in the DNA on the coding strand is the start. And if you look at the opposite, the complementary side, the complementary base pair coding to that is TAC. So if you see TAC in the template, that's also a start. It's the same thing, right? Because it's complementary to ATG. All right, but if you look up start codon, like if you Google start codon, it'll say ATG because that's the code to start. All right, so we've made that code, and it's actually at the beginning here of our mRNA as well. Now it's slightly altered because RNA does not have T's; it has U's instead. So it says AUG. Every three-letter sequence is known as a codon. Okay, CODON, codon. Because when you read DNA or RNA, you read it 3 letters at a time. And every 3 letters is an amino acid. An amino acid is the building blocks of protein. When you break down protein into its very smallest pieces, those smallest pieces are called amino acids. And each 3-letter sequence codes for 1 amino acid. So we've got a start codon, AUG, and then we've got 3-letter sequences going after that. Each one is a codon. So it goes AUG, CGG, UAC, UUA, and each one of those is a codon. Until you get to the end of your sequence, which is a stop codon, and we'll talk about what that looks like. But a codon is just a 3-letter sequence that codes for an amino acid. Now, at this point, this is transcription. We have made this mRNA sequence, should say that over the side, yeah, the message, which is mRNA. And now, this is only single-sided. You'll notice it's not a ladder anymore, or it's like a half ladder. It's only the single-sided. And so this message can actually fit through the pores of the nucleus and can make its way out and interact with the ribosomes, which are on the outside. So that's in fact what it does. It's going to exit through the nuclear pores, and it's going to interact with the ribosome. So we've created a message. We've transcribed. We've made a copy of just the code, and we've done it on the template. The template was used to make the copy. At this point, are there any questions about what we have done so far? We have just made a message to leave the nucleus, and the message is just a copy of the code with a minor variation because there are no T's in mRNA, so they're U's instead, uracil instead of thymine. I'm going to pause for a second here in case people actually do have questions, because this is, I realize that this process is not the most straightforward thing that you've ever seen. It's a beautiful day outside. Oh, and it's 17. Lovely. Oh man. Guys, after school, definitely take a moment and enjoy that fresh air. 17, sweet deal. Oh, I wonder if I get in the pool. My son will like that. Okay, I'm not seeing any questions. I will circle back if any pop up, though. Throughout this entire process, please just write your questions in the chat. Send them in. It's important to clear stuff up as soon as possible. And definitely, if you have a question, it's probably a question that other people in the class have as well. So don't be shy, guys. Okay, so we've made this message. We're going to send it out into the cytoplasm, and we're going to do the second part of this process, which is called translation. Remember, translation means to convert from one form into another. So now we're going to convert the message into a protein, a polypeptide actually first, but then that'll become a protein eventually. So we have our RNA sequence, remember, is AUG, CCG, UAC, UUA. That's the sequence we just made. And a ribosome is going to come and attach to our RNA. Ribosomes are actually also, confusingly, made out of RNA. So our RNA is really kind of cool. You can make stuff out of it as well, which is why RNA is actually believed to be the original hereditary information in organisms. This is like ancient organisms when life first formed on Earth, because it has so many potential functions. It can be a code. It can also read a code. You can like do a whole bunch of different things with RNA. So anyway, that type of RNA that the ribosome is made out of, it's also made out of RNA, is called rRNA, or ribosomal RNA. Ribosomal ribonucleic acid. And by the way, that's why they're called ribosomes, because they're made of ribonucleic acid. So they're ribosomes. A little bit confusing. So you've got a ribosome. Do I have a picture of a ribosome? Yeah, I do. Okay, so this, if you look at, let me just delete some of this stuff here to make it easier to see. So I'm going to use, I'll use the same colors. Let's say pink. Okay, so this structure, this big, what looks like a sandwich here, is the ribosome. Okay, so it actually has two parts. It has a bottom half and a top half, and it looks kind of like a hamburger. But it's made out of RNA. Okay, so it's a hamburger made out of RNA, and it comes and it attaches to the mRNA. Okay, remember that the mRNA is that message that we just created in the nucleus. Okay, it attaches and it starts reading the sequences until it comes to AUG. Okay, we already mentioned this AUG thing. AUG means start. Okay? When it hits the start, when it finds a start, and there is a small sequence that comes in front of the start, by the way, that also gets copied over from the DNA. You don't need to know any of that. But that starting sequence just helps the ribosome attach. It's not particularly important. Certainly not in grade 11. It's not. But in grade 12, you'll add a bunch of detail to this process. But anyway, the important thing here is that there's a start. Okay, that's the AUG. So it's going to read along till it sees AUG, and then it's going to use this conversion chart. I mean, it doesn't actually use a chart. This is done biochemically inside the cell, but around the outside of this chart, what you see here are amino acids: arginine, serine, threonine, lysine, asparagine, leucine, phenylalanine, tryptophan, cysteine. And those are sort of like the Lego bricks that proteins are made out of, okay? They're the smallest pieces of proteins, and they each have their own chemical properties. They have different binding sites, different shapes. And when you assemble these together, you can build all different kinds of functional pieces by assembling these in a particular order. That's the magic of genetics, which is that if you take these building blocks and put them in a particular order, you will design a structure that does stuff inside of the cell. It's kind of a miracle, really, when you think about it. I mean, it's such an amazing idea. So around the outside here, we have all the different amino acids, and you'll notice that some of them are on there more than once. And that's because there are multiple sequences or codons. Remember, a codon is a 3-letter sequence that actually code for the same amino acid. So there's a little bit of redundancy built into here, but that's how it works. Okay, so there's going to be a 3-letter code, and the way that you read this chart took a long time for us to figure this chart out, by the way. But we know the chart. We know what codons go along with which amino acids, and we've made a chart. I've put it in your notes here. You can Google these too. But you start in the center of the chart, and you read it from the center outwards. So, for example, I'm gonna use the start codon as an example. Okay, AUG is the start codon. So if we look at our conversion chart, we're starting in the center. So A, then U, then G. Okay, so I'm working my way outwards. I start with A, and that brings me to this quadrant up here. And then the next one is U, so then I go to the next one in the sequence, and then I move outward, and G is the third one. Okay, and that brings me to this piece of the pie. And you'll also notice that I added this in, but I mean, it should be written right on there. It's kind of annoying that it's not. But that sequence, AUG, is known as the start codon. Now, it's the start codon for mammals, and actually for, is it for all vertebrates? It might be for all vertebrates. But which codon is the start codon, I think, differs depending on kingdom. Now, I don't think it's necessarily exactly specific to kingdom, but these domains are not always consistent between all organisms. So what codes for what can differ. But this is accurate for, at the very least, it's 100% accurate for humans. I know that for sure. And I'm fairly certain this applies to vertebrates. I'm going to double check. But anyway, it definitely applies to humans. So there are other organisms that have other sets of amino acids and things like that they can potentially produce. So this system is a little bit different depending on species. But I'm fairly certain this is consistent for all vertebrates. It may be consistent for all animals. I'm not 100% sure. That I got to look up. But anyway, at the very least, we're only going to be talking about humans, so it doesn't really matter. This is the one that works for humans, so we care about that. So AUG is methionine. It's also the start codon. But that also means that every protein is going to start with a methionine. So a methionine is an amino acid. It's a shape. And so you are going to add that shape at the very beginning of your protein. There it is. So the ribosome is going to grab a methionine, and it's going to add it on. So you might be wondering, how does it know to put the methionine on AUG? Why does it do that? Like, why does the methionine stick to that sequence? Well, there's one piece of, that I've left out here of this puzzle, and that is that every amino acid has a tiny little piece attached to it called a tRNA. Of course, it's made out of RNA. What isn't? It's made out of RNA. And tRNA stands for transfer RNA. And that little piece, it looks like a little clover kind of on the end of it. I didn't draw it very well here. It actually does kind of have like a clover shape. That's what it said anyway, that it looks like a clover. And it has a section on it, and that section is actually right at the very bottom of it, right down there, that will stick to the specific sequence on the mRNA. So if you go back to our ribosome here, you'll notice here's the ribosome, the mRNA getting fed through. And there's a sequence right here, this sequence right here that's currently being read, CAG. Okay? And then here's the tRNA. That's this thing. This is the tRNA. I guess it doesn't really look like a clover. I don't know what to call that. A T? T-shapes, maybe? And it has the opposite, again, the complementary base pairs on it. So those complementary base pairs are going to stick to this section of RNA, and they're going to deliver the correct amino acid to the sequence. So once that happens, that amino acid then is going to be bound, stuck to the protein that's being formed. Here's the chemical bonds being formed here. That's meant to imply chemical bonding. And then it's going to get stuck to this growing ring, or not ring, growing string of amino acids, okay, which is eventually going to go on to become a protein. Okay, so we're going to stick on more and more of these amino acids and then build out a long string off of it, okay? And then the tRNA, the bottom part here, is actually going to break off. It will find another amino acid and then it gets used again. Okay, so the tRNA keeps getting cycled and cycled and cycled. But that's how you actually get the right amino acids to stick to the RNA and to actually build this protein in the correct order. Okay, seems like a lot of information there, but basically what we're doing is we're taking the code that we've made, the message, and we're reading it 3 letters at a time, and for every 3-letter sequence, called a codon, we're going to add another amino acid. So we've gone AUG, we added a methionine. Next it goes CGG. Okay, so let's look up at our code reader here. CGG. Okay, remember reading from the center outwards. CGG is arginine. And there's short forms for these, which I'm going to use all the time. They're on the outside. But arginine is ARG. And then so the next one is arginine. And then next is UAC. So again, we're just start in the center. UAC. And that brings us to tyrosine, which is short form TYR. And then there it is right there. So we added in tyrosine. And next, we have UUA. And you go over here and read it off. And it goes UUA. And that brings us to leucine. So then the next amino acid that's going to get attached is leucine. And on and on and on it goes until it reaches a stop codon. So you'll see them on the outside here. They're actually labeled stop. Let me highlight them in yellow. So here's a stop down here. Here's a stop here. That might be the ones for humans. So for humans, it's UAG or UAA or UGA. Those 3 sequences say stop. And that basically, when the ribosome gets to a stop, instead of adding an amino acid at the end, it lets go of the protein that it's building so that it can float off and be, sometimes it's usually modified after that by some other organelles, which we're going to talk about in a second. But at that point, it's done being assembled, like it's done being put together. Okay? So just by building the amino acids in the correct sequence, they will stick to each other in a certain specific way, and then you will end up getting a complex shape out of it that does a job. It has a function. It's a signaling molecule, or it's structural, or it builds something, or it's an enzyme, so it causes chemical reactions to happen in the cell. Proteins do all these different jobs, and it's all built from this chemical process, really. This is all chemistry. It's biochemistry, but it is chemistry. So anyway, you can see how understanding this process, how basically everything is made in all living cells in nature, is pretty much like the greatest discovery ever made in biology. I mean, this is how life is assembled. So, and again, why it's called the central dogma. It's the most important concept in biology because it's about how all living things are assembled from molecules. So it's pretty important. At this point, before I move on, I'm going to talk about what happens after this process now. Once we've made this string of amino acids, I'm going to talk about just some kind of sort of final touches about where the proteins go before they're done. But before I get to that, is there any questions about this process? The process of transcription, which is making a message out of RNA, or translation, which is taking the RNA, reading it with the ribosome, and making an amino acid sequence, or what will become a protein. It's really a polypeptide here, but I'm kind of splitting hairs. I'm gonna, you know what? Oh, time for a poll. Let's do a poll. What? I'll get faster at this. Okay. Answer this poll. All I'm asking here is how comfortable are you with the idea of this so far? I want to have a quick look at how the class is feeling about this before we go on. I realize this is a complex process, so I want to know if I need to circle back on anything. Remember that this poll is anonymous, so you don't need to feel embarrassed. No one's going to see your results or anything. This is a good tool. I'm gonna do this more often. Okay. That's most people that have responded at this point, although not everybody. I wonder if I've lost some people since we checked in for attendance. But may I ask, for the people that have said that they would like more clarification, if that is the kind of thing that I could provide, it's more than one person, by the way, if that's something that I can provide now, would you be open to letting me know which area of this you would like more clarification or examples of? And if not, that's okay. Again, I'm not trying to call you out. This is anonymous intentionally. But if you would feel comfortable, if you wouldn't mind putting in the chat where you would like additional clarification. It is a few people. If you're not comfortable, which is totally fine, please consider stopping by during the Google Meet so we can just have a quick chat. And I'm very confident that I can get you up to speed so that you feel comfortable with this. We're going to do some practice together, so we're not done with this concept. But if you're really feeling unsure at that point, during the meet, please consider coming to talk to me. For everybody else, which is the majority, I'm going to move on. I think we should be okay to go on then, unless I'm going to get a specific question. It's a little bit hard for me to pick out exactly what that person or what those people would like me to elaborate on. So I'm gonna keep going for now. So what I want to mention is, when this is done, okay, so you've done this process, you've built your chain that's coming out here. Oftentimes this chain, by the way, will do some folding and it'll stick the amino acids are going to stick to each other in like a unique pattern. And when they do that, and this is three-dimensional, by the way. I'm showing it two-dimensionally here, but it's a three-dimensional object being produced. That is going to form the functional protein. Okay, so there it is. Boop. A sequence, an amino acid sequence is done. It gets released, and it becomes a protein in the cell doing its job. Okay, so that's one route. The protein could just be done at this point. If the protein is done at this point, that is what's called an intracellular protein. Intra, the prefix intra, means within. And this is not just in biology. There's lots of areas where that prefix is used. But just so you're aware, whenever you see intra as a prefix, it means within. If the protein is done being built at that point, it is an intracellular protein. And intracellular proteins are proteins that are just going to be used in that cell. And that happens. Lots of the proteins that cells make are for their own cells, like for within that cell. And so it becomes an intracellular protein. And those ones are made by the free-floating ribosomes. So you'll remember from the cell organelles stuff that some ribosomes are just floating around in the cytoplasm. Floating ribosomes, or they're called free ribosomes that aren't attached to anything, make intracellular proteins. There are also ribosomes that are attached to the endoplasmic reticulum, which you're also aware of. That makes the rough ER, right? Those ribosomes have a different job. They make intercellular proteins. So those are proteins that are going to be exported out of the cell. So they're for the organism to use. And obviously, a lot of proteins are for external use from the cell. Digestive enzymes, transmission hormones. If you want to send a signal to another cell, that's going to be exported. So there are lots of intercellular proteins as well. Inter means between. So an intercellular protein means a protein that is for between cells. Okay, so it's going to exit the cell. So I mentioned this at the bottom here. There's sort of two routes. Free-floating ribosomes produce intracellular proteins. That's for within the cell. And rough ER ribosomes produce intercellular proteins. That distinction between inter and intra is very, very often confused by students in this course. I mean, probably in students in biology courses everywhere because their words sound very similar. But that's something that you're going to want to file away and make sure that you don't confuse because they have completely opposite meanings to each other. So inter and intra are very different. Intra means within, and inter means between. Silly science terms. Why'd you make those sound so similar? That's annoying. If we're talking about intracellular proteins, those are the ones that are going to stay in the cell. What I've just described to you, that process is the whole thing. They're done. The proteins are done at that point. And you would call what comes out of the ribosome at that point a protein. It's done. It's been folded and it's going to be a functional unit. But many proteins are not complete at that point if they are intercellular proteins. That means that they're going to go outside of the cell. They usually have to be modified, snipped, folded. Adjustments are made to the protein. Sometimes they're built into multiple subunits put together and stuff like that. I mentioned it on the next page too. I really, I'm harping on this a lot because this is just something that students confuse a lot. So what I'm going to talk about here now is the process here for an intercellular protein, one that's going to leave the cell. So there's just a couple extra steps that you need to know. Okay. So first of all, I've already mentioned here the first part, step 1, which is that ribosomes, but in this case they're going to be rough ER ribosomes, right, are going to read the RNA. Okay, so the RNA attaches and it builds a sequence of amino acids. Okay, so the mRNA is fed through the ribosome. This looks exactly the same as what we already talked about. The RNA, remember that's the message, is going to go through the ribosome. It's going to get read 3 letters at a time, a codon, 1 codon at a time. And you are going to create that string of amino acids. That's the same. But in this case, it's not finished once it's built. So we don't usually call that a protein at this point, because a protein is like a completed, folded string of amino acids. So what a better word to use, and I noticed, by the way, that the original note here, which I got from another biology teacher, we share materials all the time, but in the other note, they called it a protein. It's not 100% correct to call this a protein, so I'm not going to call it that. I modified it. It's not entirely wrong. It is protein-like. It's an almost protein. But really what it is a bunch of amino acids stuck together. And there's a word for that. So when you stick a bunch of amino acids together, but they're not done being folded or modified, that's called a polypeptide. Poly means many, and peptide means amino acids, basically. It means of protein, but it's not quite a protein yet. So a polypeptide is like many of the components of a protein. You've come across the prefix poly before in lots of places. Polyatomic ions, polygons. It's a common term, or a common prefix. Okay, so we have a polypeptide. Okay, then the newly created polypeptide, not protein, moves through the rough ER. So the rough ER is actually going to transport that polypeptide, okay, and pinch it off in a little balloon. Okay, that little balloon of cell membrane is called a vesicle. And it really is just a little balloon inside the cell made of cell membrane. So you're going to create a little pocket, and you're going to pop your polypeptide inside the pocket. The purpose for that pocket is just to transport the protein around. And when you're doing that, you want to keep it separate from the other stuff that's floating around in the cytoplasm. It's basically just like if you were going to transport something from one house to another, you'd put it in your pocket, as opposed to just dragging it along behind you, where something could happen to it as you drag it along. You just put it in a pocket. And that's what the vesicle is. You just pop it in a little pocket, and then its next stop is that it gets sent to the Golgi apparatus. Okay, this is where the Golgi apparatus is going to come into play. And you guys reviewed already the function of the Golgi apparatus. And I believe the function that's stated there is that it packages proteins for transportation. Or at least that's one of its main functions. And that is, in fact, what it does here. So you're going to give it to the Golgi apparatus. And the Golgi apparatus is going to take the polypeptide and fold it. It does some of the finished folding of the product. Okay, so it folds it into a 3D complex shape specific to the function. All right, so it needs to be a particular shape in order to get it to be functional. Now, once you fold it into its finished shape, now it's called a protein. Okay, because now it's a functional unit that will do a thing that you want it to do. So that's a protein at that point. If you unfold the protein, that's called denaturing the protein. It doesn't function anymore, obviously, if you unfold it. And then it really goes back to being a polypeptide. Again, I'm kind of splitting hairs here, but later on when we talk about digestion, we're going to talk about the opposite of this, which is taking a finished protein and then unfolding it and cutting it up which is what you do when you digest protein. And then it doesn't really make sense to talk about it there, but not talk about it here. Because we're calling it one thing in one lesson and a different thing in a different lesson. That doesn't make sense. So I'm trying to keep things consistent here between the units. Once you've folded it up, the polypeptide, then it's a protein. Okay? And it gets repackaged. So that's the second part here, is that the Golgi is going to put it in a brand new bag. Okay? A vesicle. And the reason why you want to put them together in vesicles is because you usually want to release the proteins into the cytoplasm. Remember, these are intercellular. They're going to leave the cell. You want to release them in groups, usually a bunch at once. So for example, in your pancreas, when you eat a bunch of sugar, you want your pancreas to release insulin. That's what it does, so that all of your body cells can get the signal that says, okay, take up the sugar. There's lots of sugar. Take some sugar. There's sugar available. When your pancreas cells release insulin, they don't just release like one insulin per cell. Every pancreatic beta cell, which are the ones that make insulin, releases a bunch of insulin. Okay, big packages of insulin, and those packages are made by the Golgi. Okay, you can also store those packages in the cell, ready to ship them out, which is again a useful thing to do, is to kind of get them ready in advance and have them sitting waiting in the cell, which is another reason why you're basically putting them in a crate for transport. Okay, you're packaging them all together in a crate. Okay, now it's a protein. When that protein is required by the body. Okay, so now when we get to the point where we're saying, all right, send out the blank. Send out the testosterone. It's puberty time. Okay? Or testosterone in the blood is getting low. Send out additional testosterone. Okay? A signal comes in saying release the protein. Then that is when the vesicle is going to pinch off from the Golgi. Okay? So it'll pinch off, and then the protein gets sent to the cell membrane where it's going to be exported from the cell at that point. The vesicle is going to merge with the cell membrane. That is, it actually becomes part of the cell membrane. And when it does that, it basically blows all of its contents out into the intercellular fluid, which is the liquid that is outside of the cell. Okay? Or you could say that it dumps its contents to the outside of the cell. And usually, depending on where this signaling molecule is headed, it's usually transported in the circulatory system. So that extracellular fluid is actually continuous with the blood. So that liquid and the blood are the same liquid. So it's going to go from the intracellular fluid into the blood flow, and then the circulatory system will pump it around the body, typically. Sometimes those chemicals are only meant to travel a couple microns, teeny tiny little distances, 2 micrometers or less. Maybe they're meant to travel 1/10 of a micrometer or something like that to a neighboring cell. That would be in the case of something like a neuron, a nerve cell, communicating with its adjacent cell, with the cell right next to it. And then that little distance in between is called a synapse. And in that case, the distance that it has to travel is like absolutely tiny. It's a tiny, minuscule little distance that the protein has to travel. So it's not going to go on a long journey in the circulatory system in that case. But most of the time, often, extracellular proteins have to join the circulation, get pumped around, and there is a slight delay. It's not that long of a delay. If you've ever, I'll give you a great example of how rapid this process is, because it's actually surprisingly fast. If you've ever gone on a roller coaster or done something where you've, maybe scared is the wrong word, but excited yourself, felt a thrill, okay? And there's lots of things that do that for humans, but you gone in a haunted house or you watched a horror movie and you did something that was, made your heart race. Okay, that's actually a hormone response It's initiated by your adrenal glands, which are sitting on top of your kidneys, and they are dumping using this mechanism. So the Golgis are sending vesicles with adrenaline out into your circulation from your adrenal glands, which goes into your circulation and activates your fight or flight response, which causes your heart to race and you feel that flutter when you get scared. And that's very rapid. Imagine how quick that is, that you like see something scary in a horror movie and your heart is racing. I mean, what's the delay there? Half a second? That's how quick it is between when you initiate that release of a protein neurally. In that case, your nervous system is sending a signal to your adrenal glands to release it, which is that part's fast. But then the actual releasing, like moving these vesicles to the surface of the cells and releasing them into circulation, I mean, it's taking fractions of a second in order to do that. So I find that so fascinating. These systems are very efficient. They work so well. And then in a very short amount of time, you've released these chemicals into the circulation, and they're already functioning. They're already binding to receptors on your heart and increasing your heart rate. And, imagine how quick that is. It's so fast. So it always fascinates me how quickly this stuff actually works. What's mentioned at the bottom here is just this idea of the fact that if the cell itself needs a protein, so that's an intracellular protein, that is just the free-floating ribosomes that do it. So step number 7 and 8 here are basically just like, what if it takes the other pathway? Well, it's going to do these things, okay? So it's just free-floating ribosomes that are going to do it. And then it enters the cell's cytoplasm and starts doing its function inside the cell. So it just eliminates a bunch of steps in that case. So one of the... oh, did I remove that activity? No, I didn't. Part of your homework for tomorrow, and you can do this at any time. Ideally, it would be like during Learning Block 3, but you can do it whenever you want, is what I'm going to get you to do is have a look at this picture. This is, by the way, I skipped over a page. We're going to go back to that page. But I just want to mention this activity. So what you see on this page is a nucleus, a rough ER, a Golgi, and then right at the very end at number 4 there, you see a vesicle releasing proteins into the extracellular fluid. What I'm going to get you to do is write out the process that we just learned, all the way from you have a piece of DNA, all the way to you expel protein out of the cell. Okay, step by step. I want you to write it out in your own words. And you can use scientific language. You absolutely feel free to use the vocabulary that you've just learned. But I want you to try to write out the entire process, what is happening from beginning to end. And the purpose of doing that is just to help you synthesize and cement this information into your brain. Okay? Reading it over, it's very easy to just read this and be like, yeah, I get it. It does this thing, it does this thing. But if you have to go through and describe what is happening for every step, you will have to think about it. You will have to think about what to say about each step of this process as you go from double-stranded DNA to making a single-stranded mRNA to using that and decoding it with a ribosome and reading 3 letters at a time, which is called a codon, and producing the correct amino acid sequence, which is then going to go travel through the ER and be folded in the Golgi and repackaged into a vesicle. And if you have to write that out yourself, you are going to have to think about it. And that will really help cement it in your brain. Honestly, these writing activities are super helpful. They seem labor intensive, but if you want to commit a process to memory, it really helps to try and write it out. I mentioned here that you should use proper sentence structure and stuff. You can do that if you want. I actually, again, we share these activities between us, but I don't focus on that stuff personally. Do I think that it matters when you're writing out this process if you use point form or whether you write it out in full sentences, like with proper sentence structure? No, I personally don't think that makes any difference whatsoever. I mean, the phrases and stuff that you use in your point form have to make sense, but I think it's perfectly fine to do this in point form if you want. So that's where I differ from some of the other science teachers. Ooh, I shouldn't record this on video, otherwise they'll find me. But I'm not really I'm not gonna care about that too much. What I'm really looking for here is that you make an attempt to actually write out the steps one after the other, okay, in sequence. I've given you some ideas here for how you could break it down. This is I sort of labeled this here as a 4-step process, but I mean, obviously it's more than 4 steps. So each one of these steps has like many sub-processes that take place at each of these various locations. If you want to add in steps, that's fine too. But consider breaking it down into steps. That's essentially the important part here, because it'll make it easier for you to recall and understand, and easier to summarize if you break it down into steps. Okay, sorry, Bree. Everything that we've been writing directly in this booklet, should that be in our homework document for this unit? No. So I'm not checking this. The way that I'm evaluating this process is going to be on the quiz. I'm going to evaluate this on the quiz. Should you be doing these activities even though I'm not collecting them? 100% yes. If you're not doing these activities, you are going to struggle with this. And I don't say that gleefully or anything, but but it's in your own best interest to do this. I don't believe in busy work. I don't believe in getting students to just do stuff like for the heck of it, because it's just like it's something to do. Like either the activity has a purpose in learning something, or it doesn't. I'm giving you these tasks because they are helpful in actually committing these processes to memory and understanding them correctly. So I mean, again, you could choose not to do it, but you're in a biology course for a reason. This is the most important concept in biology. So if you're not interested in having a good understanding of how this process works, what are you doing here? You've misplaced yourself. Sorry, and that's not directed at anyone in particular. But honestly, like when it comes to education, especially now where you guys are, grade 11, I mean, at this point, you're picking stuff that you're interested in for interest's sake, because you will need it and because it's intriguing. If it doesn't meet that standard for you, take a different course. Take a different course. Like these are life decisions that you're making here. So my assumption is that you care, and that you will do these activities because they are important in order to learn these processes correctly, and you need to know that for literally anything you're going to do in biology. So my assumption is that you'll do them. Again, you can choose not to. That's fine, but you won't do very well. So it's up to you. But no, I'm not going to collect this. I also don't evaluate your homework for correctness. What I'm evaluating there is your learning skills, whether you're making a concerted attempt to complete the work, because that's in your best interest to do that. And those learning skills are probably what will serve you over and above what you actually learn in this course. A lot of stuff that you're going to learn, this is not one of those things, by the way. This is actually really important to understand how this process works. But a lot of stuff you're going to learn in this course you'll probably never use again, and that's fine. But the learning skills, doing the work, learning how to commit a process to memory, understanding vocabulary, that will serve you no matter where you go in science. Even if you don't even stick with biology, in any science, that's going to serve you well. So, I mean, learning how to properly research a subject, learning how to present your research in an organized way. These are skills that are like absolutely cross-platform here. You're going to use that in every discipline. So, again, it's not a time-wasting process here. This stuff I learned in my grade 11 biology course, I've been using my entire life. Now, I realize that I'm a biology teacher, so I'm absolutely going to use it all. But even if you don't specifically go into biology, I could pull my wife down here. My wife's a family physician. I mean, she uses bits and pieces of this literally every day. So, I asked her about angiotensin. That's, one small example, but like understanding how hormones are produced and distributed. And again, this is in medicine, something you must know. So, or nursing or whatever. I could name a million things, but, anyway, I don't believe in busy work. When I'm asking you to do something, it's not to kill time. It's always for a better understanding of this process. So I highly recommend this, I guess, is maybe another way to phrase it. But try this. It'll help. Okay, the other thing that I'm going to get you guys to work on for the next hour or so. And by the way, I realize that... Ooh, whoops. What? Pop-ups blocked. I realize that... where are we here? Ooh, mitosis review. Ooh, I was supposed to get to that today. How are we doing for time? Ooh. We're definitely not going to get to meiosis. That's super complex. We'll save that guy for tomorrow. But ooh, I do have to do this though. Okay, I'm going to stop talking about this and very quickly describe what this activity is, and then I'm going to kind of push that activity towards the end because I do have to do a quick review of mitosis as well. So sorry, my apologies. I've been extra verbose today. That's a real bad habit of mine. What you see on this sheet is practice in decoding DNA and understanding how to build an mRNA from a DNA. Okay, so I'm going to do the first example with you. Let's get rid of all this. Shoot. And this is available in the course notes that are posted online, so you can find this and take a look at it if you want to check your answers later. But get rid of all this. So what you're doing here on this sheet is you are practicing creating a protein from a strand of DNA. So what I give you in each of these cases, well, some of them actually, hold on. You actually have to figure out in a bunch of these cases. But what I'm giving you in the first case anyway is the template strand of DNA. So I haven't given you the code. What I've given you here is the template. Okay? Now, I have been using the setup here where I've always put the template on the top and the coding on the bottom in all the examples. And so I'm going to do the same thing here. I'm going to get you to fill in the coding strand underneath the template. Okay, so remember these first two parts here, the top two, are the two sides of the DNA. Is this going to work? Okay, so that's the DNA part, the two-sided DNA. So I'm going to go along here and just simply write what is complementary to the template, and that would be the code. So the code, remember, is the actual information that you want to be the same, make your copy of. Okay, and it's going to be complementary to the DNA. So A is complementary to T. Maybe I'll use red here. Actually, I'm going to use red because we used green for the mRNA before. Maybe I'll try and keep that consistent too. So A is complementary to T, T is complementary to A, C complementary to G, C is G, G is C, C is G, T is A, C is G, C is G, G is C, C is G, G is C is G, okay, etc., etc. So what we have here at the top is the DNA. It's double-sided, and there is a template, which is I'm drawing on top, and a coding strand, which I'm drawing on the bottom. Okay? The mRNA strand is going to be built on the template. That's what the template's for. But it is the same code as the coding strand, except all T's are now U's. So in order to build this, you need to look at the template, and you're going to build the complementary sequence to the template. Okay? So complementary to T is A. And yes, it looks the same as the code. That's the point, right? We're trying to make a copy of the code. So it's going to look the same as the code. However, anytime that you would put a T, you're actually going to put a U. Okay? So C is complementary to G, G is complementary to C, C is complementary to G, T is complementary to A, C is complementary to G, C is complementary to G, G is complementary to C, C is complementary to G, C goes with G, G goes with C, T goes with A, C goes with G, G goes with C, A goes with T, but there are no T's in mRNA, so it's going to be U. C goes with G, A goes with T, A goes with T. Okay, on and on. Okay, you go through. You're going to make your mRNA. Then you're going to look at it 3 letters at a time. And I've tried to organize these 3 letters at a time to make it a little bit easier. I've kind of broken them up a little bit. But remember that 3 letters at a time is called a codon. And then you're going to go through, and the AA at the bottom here is the amino acid sequence. And in order to figure out the amino acid sequence, you got to go through and read the decoder ring, basically. So AUG is our first one. I'm going to just skip back here. Ooh, this is going so slow. I'm going to have to restart this. Once it starts going this slow, it's about to crash. So I'm going to restart this fast. Sorry, it's just a bit of a pain. There we go, much faster. Okay, so here we go. What was it? AUG. Okay, so we look at our decoder ring. You guys have a decoder ring too. You may want to open one of these mRNA codon charts or protein building charts. You can find them pretty easily. Just Google it. Google image search. And then you just can have it open in a different window if you want. Like you can organize your tabs, like have one tab on the left and the other one on the right. It might be easier to look back and forth on this thing. Or if you can print it off, that's an option too as well. Anytime you would need this on the quiz, I give you the chart. I just put it right in the quiz question, so you don't have to look it up because that's kind of unfair. But I do ask you to do some of this decoding in the quiz as well. So it's AUG was the first one, AUG, and you'll notice that is, of course, the start codon. It is at the beginning. So, and it also codes for an amino acid, which is methionine. So MET is the short form. Short forms are fine. I'm going to write that right down here. MET, that's the first amino acid. Okay? And now we got to decode the next codon, which is GCG. So we go back to our conversion chart. Go GCG. We're reading from the middle outwards. And then that codes for alanine, ALA. So we go down here, and then we write ALA, that's alanine, and then we move on to the next codon. Okay, so you're going to go through and you are going to figure out the amino acid sequence for that sequence of template. Okay, you're also going to add in the coding strand. It doesn't actually ask you on the original sheet to write the coding strand, but I personally find, for understanding reasons, it's better for you to add that in there so that you understand that there is a coding strand that is attached to the template that has the actual code on it. That's the strand that has the code. So I'm going to get you to. I just added that part in, but you should put that there as well for each of these examples. Okay. So for the part below, right? Great question. Great question, Bree. So for 2. There are also, so here you're going backwards, right? You have the mRNA and you go backwards and you make the DNA. It works exactly the same way. So the template on the top, code on the bottom. Remember that the code is basically the same as the mRNA, and the template, which is on top, is the complementary to the mRNA. And then you go through and you do the amino acid sequence. That's exactly the same. For number 3 is a little bit tougher because I'm giving you the amino acids, and there is actually more than one code that could make that amino acid. Now, for the very first one, there's only one code that you can use because you have to use a start codon at the beginning. Okay, Bree? So the very first one has to be the start codon. It has to be AUG. AUG is the start codon. Okay, that's AUG. You have to use that one at the beginning. There's no way around it. However, there are multiple versions for the rest of these. So for example, there's more than one code that makes arginine. So AGG is one of them, but there are other codes that also make arginine. So you use whichever one you want. Okay? It may not look exactly the same as mine, but that doesn't matter. As long as you've used a code that codes for arginine, it's correct. Okay? So this goes, there's a question like this on the quiz, but the way it's set up on the quiz, there's only one answer that makes sense. The other ones don't make sense. But in reality, it could be any of them. So if you want to build an arginine, it could be any one of the 3-letter codes here that codes for arginine, and they're all the same. So the sequence of DNA may be slightly different in your answer, but as long as you've used an mRNA that codes for arginine, it doesn't matter. You've done it correctly. Okay, so just something to be aware of if you're checking your answers for number 3. or 4, for example, because there are some amino acids that are given in number 4. For those ones, when you're converting backwards from amino acid, there's more than one sequence that could potentially go there. So it may not be exactly the same as the sequence that I've put. Okay? So anyway, I'm going to get you to try these. There's a couple sort of different ways to do this. In other words, you're either going from the top to the bottom, you're starting with DNA and making an amino acid exactly the way it happens in real life, or I've given you the mRNA and you're telling me what the DNA and the amino acids would be. And by the way, problems like this come up in science all the time as well, where we can see an mRNA that is being created in large quantities, and we want to say, okay, well, what's the DNA sequence that is being read? What is the amino acid that's being made from that? I mean, we do that all the time in biochemistry. So there's value to being able to do these as well. Okay, so I'm going to get you to try that. Once you go away and do your own stuff here for that. So there's this, and then I am also going to get you to complete this transcription and translation piece, okay, where you're going to attempt to write out the process. Before you get to that, I'm very quickly here going to review mitosis. Okay, normally I probably spend a little bit more time on this, but we're just a touch behind. The reason we're reviewing mitosis is not because mitosis is important for this unit. It's not important on its own. The reason we're reviewing it is because we're going to talk about meiosis tomorrow, and it is very similar to mitosis. So if you have a good memory of mitosis, you recall how it works and what the stages look like, it will make it much easier to understand meiosis, which we're going to cover tomorrow. So I could just do meiosis and say, remember last year? Do you remember interphase? Remember prophase? Remember metaphase? But I'm going to just take a guess and say the answer to those questions is no. You don't remember prophase and you don't remember interphase, because why would you remember that? I think it's unlikely. So what I'm going to do here is do a quick review of mitosis. I believe that there's a video that you can watch here that I've attached as well for an additional review if you'd like it. But you don't need to watch it if you don't want to. But anyway, what's the purpose of mitosis? Hopefully you guys remember this from last year. You want to make an exact copy of your cell, right? That's the point. And this comes up all the time. You have one cell, you want to make 2. You want to make some new tissue. You want to make a new, whatever, repair yourself, growth, whatever. You want to make a new cell. So in order to do that, the most important part of mitosis, and of course, just based on what we've just been talking about, is of course the DNA part. We want to make an exact copy of the DNA and put it into each cell. If you don't do that, both of the new cells that you're going to make are going to cause problems. So you pretty much have to make an exact copy of the DNA, because that's the instructions for building all the stuff. And if you don't do that, nothing's going to work. So I really focus just on the DNA here. Now, of course, you make copies of everything. You make a copy of the Golgi, and you make a copy of the endoplasmic reticulum, and you make a copy of, this and that. Yeah, sure, that's important. But it's not nearly as important as the DNA. So I'm really just going to focus in on the DNA part of this. You may remember from last year that most of the time a cell is just hanging out in interphase. Interphase is just when the cell does its cell stuff. If you're a nerve cell, you're conducting nerve signals. If you are a beta cell in the pancreas, you're making insulin. If you are a skin cell, you are acting as a protective barrier. If you're a melanocyte in the skin, you are making melanin, which is protecting the surrounding tissue from UV damage. Whatever, you name it. You're just doing your cell job, okay? And there's 3 parts to interphase. Most cells in your body, by the way, are in interphase because it makes up the vast majority of a cell's life cycle. And most cells are just going to be in that phase naturally. But you got to make copies sometimes. So there's 3 parts to interphase. G1, gap 1, which is when the cell grows and makes proteins and builds stuff. It's like growth, okay? Part 2, which is synthesis. And that's when you make a copy of your DNA. Now, for our purposes, that's the most important part of interphase, the part where you make a copy of the DNA. Okay? So that happens during the synthesis phase, during interphase. And then the last part, you start making structures that are useful for dividing. Now that's important. You make centrioles, which are going to make spindle fibers later. You might remember this from last year. But again, for our purposes, it's not that important because it has nothing to do with the DNA. But you do make centrioles during that part, okay, which are going to be used to move the DNA around. Then you get mitosis. So mitosis is that very short part, the mitotic phase, where you actually separate out the DNA into two new nuclei. And then right at the very end of the process, you get cytokinesis, which is essentially just splitting the cell in two. Okay, and then that's the cell cycle. Then you go back into interphase, and then you go through it all again. Okay. During the S, we mentioned this. During the S phase, you need to make a copy of everything, right? So what happens? You remember the karyogram from earlier, or the karyotype. Here are the 46 human chromosomes. Okay, you have 46. What happens during interphase is that you are going to make an identical copy of each of these. So remember that each one is considered to be a homologous pair. Okay, we have homologous pair 1, homologous pair 2, homologous pair 3, homologous pair 4, and I've labeled them blue and pink here just for maternal and paternal. Okay, so you have a dad version and a mom version of each of these, from your mom and from your dad. After interphase, after the S phase, which is synthesis, when you make a copy, remember synthesis means to build, you are going to have now a sister attached to each of those. Okay? So there will be 2 paternal and 2 maternal for each chromosome because you've made a copy of each of those. So now you have still homologous pairs. Okay, the blue and the pink here are homologous pairs. However, there are also copies of each that are attached, and those are called sisters. Hopefully this terminology is ringing a bell for you here. Sister chromatids. I'm going to let that sink in for a second. So we have now sisters for each previous chromosome, which are identical. The homologs, that is the blue and pink here, are not identical to each other. They have the same genes, so they code for the same things, but they're not identical because one's from your mom and one's from your dad. But the things that are the same color here, like the 2 blues and the 2 pinks, I'm talking about the ones on the left here, the 2 blues and the 2 pinks, are identical to each other, and they are called sisters. So the number one thing that tends to confuse students about this process is the difference between homologs and sisters. And they tend to confuse those all the time, which makes this way, way harder. So remember that a homolog is these. You have one chromosome, one from your mom and one from your dad, and those are called homologs or homologous pairs. But then you make a copy of them. Okay, that happens during the S phase, and now those are called sisters to each other. The 2 blue ones are sisters, and the 2 pink ones are sisters to each other. But the blue and pink here are still homologous pairs. They're homologs. Okay, hopefully that's okay. That's basically the whole thrust here. All that it says at the bottom here is that because you've made a copy of everything, you now have 92 total pieces. Okay, you don't say that you have 92 chromosomes because chromosome implies unique genetic information. So you've made a copy of 46, and because it's an exact copy, the 46 is an exact copy, you didn't double the amount of information. It's just like having 2 books that are identical sitting on top of each other. It's not double the amount of information. It's just the same information twice. So you still have 46 chromosomes at this point, but there's 92 chromatids. Chromatids are pieces of DNA, okay? Once they go into chromatid form. So once they're wound up, their DNA is wound up into that form where you can see it under the microscope. That's called a chromatid. So you have 92 pieces. All right, okay. So you've made that duplication, and now you're ready to go into prophase. Okay, so all that has happened during interphase. You've made your copies like this, and now you're going to go into prophase. So just to recap very briefly here. How am I doing for time? Oh, we're going to make it. We're going to make it. That's exciting. I thought we were going to run out of time, but we're actually fine. Okay, during interphase. Ugh. The first part is growth. Going to grow, produce organelles, make proteins, do your cell job. The second part is synthesis. So that's what's being shown over here. Okay. Doubling the DNA. And then the last part, you're going to build some centrioles and a couple other things that you don't need to know unless you're taking a really advanced biochemistry course. So in this case, centrioles is good enough. So remember that these cells have the full complement of genetic information for an adult organism. Okay, that is diploid. And as I mentioned here, 2N, that's how you show diploid. Both of these cells are considered diploid, both before and after you double the DNA, because you still have only the same amount of information, 46 chromosomes worth of information, even though you have twice as much material because you've doubled it. It's an exact copy. So you would call both of these diploid. Technically, this one has double the 2N number in it, but you wouldn't call it 4N. 4N would mean tetraploid, and that doesn't happen. So it's not it's not double the amount of information. Okay, and now we're ready. But keep in mind that the DNA is still chromatin at this point. It's unwound. You can't actually see it under the microscope. Okay? Then in prophase, we're going to take that chromatin, okay, which is a long noodle of DNA laying out in the nucleus, and we have to, in order to organize it, we're going to zip it up, wrap it up into a tube of DNA. We wind it around these little tiny proteins called histones, which you don't need to know for this course. But it gets wound up into a DNA sausage, for lack of a better word. And if you're in my grade 10 class, I already call them DNA sausages. That's really what they are. I mean, it's like you're taking a bunch of long string of DNA and just like packing it into a tube. So you got these DNA sausages, and now they're called chromatids. All right, so that's part of it. Chromosomes condense and become visible. That's the chromatin becoming chromatids. The cytoskeleton, which is basically like a bunch of proteins that make the cell take a certain shape, have to get broken down because they're in the way. So if you can imagine, if the cell is full of all this scaffolding and proteins and stuff that are kind of making the cell have a certain shape, you have to get rid of that because all this DNA and stuff is going to get caught on it when you try and move it around. So you bust up all that cytoskeleton stuff, get rid of it, and you start building spindle fibers. There they are at the top there, which are eventually going to grab onto pieces of DNA. And the centrioles, 2 centrioles is called a centrosome, by the way, are going to migrate to opposite sides of the cell. And that's in order to, eventually, it's going to pull the DNA to them. So you want some of the DNA to go one way and some of the DNA to go the other way. So you got to put the centrioles on the opposite ends of the cell. And you got to get rid of the nucleus. Again, the nucleus is in the way. We normally keep the DNA in the nucleus to keep it safe and separate from all the other oxidative processes and stuff that are happening inside the cell. But for this, we have to get rid of it because it's in the way. So we got to bust the nucleus up. Hopefully at this point, as you guys are all listening to this, this sounds relatively familiar to you. This is one of the core concepts of grade 10, of the biology unit in grade 10. So I'm hoping this isn't like, I've never heard of this before, because that's probably bad, but hopefully not. So all I've labeled on the left, or on the right-hand side rather, is basically pictures of this happening. The reason I color-coded it is just to show that in the X, the two sides are identical to each other. I made them the same color just to show that they are sisters. They're identical. The reason why I harp on that, by the way, is because when we talk about meiosis tomorrow, a bunch of other stuff is going to happen that it makes it really important that you know the difference between sisters and homologs, because both of those things are going to interact at some point in meiosis. It doesn't happen here in mitosis, so it's not as essential, which is why no one even discussed it with you in grade 10 probably. But it's going to be extremely important for meiosis. Okay, so we've done all that stuff. Now in metaphase, we are going to attach some spindle fibers to the centromere, which is in the center of each pair of sisters, and we're going to pull them into the center of the cell. There is a plate of protein that lies in the center of the cell called the metaphase plate or equatorial plate. I prefer metaphase plate because it's easier to remember because it happens in metaphase. And each sister is going to sit on opposite sides of the plate. The plate is basically where the cell is going to split into two. And so we want one of the sisters to go into one of the daughters, and the other sister chromatid to go into the other daughter. So we're going to line it up right on the plate, so one goes one way and one goes the other way when we yank on them. Whew. I don't usually talk this much. This is more of a talky day than normal. I feel like that's partly my fault that I'm being over verbose. Okay. Anaphase, which I like to call banana phase. It is when the X's get pulled apart and turn into bananas. That helps me remember what happens in anaphase. I don't know, may not be helpful for you, but I call it banana phase, which has the word anaphase right in it. And that's when the sister chromatids are going to get yanked apart. Now, I mentioned in the background here that you can see that there's a little cleavage furrow, which is when the sides of the cell start to poke in a little bit right here. And that is the very beginning of cytokinesis happening. Cytokinesis is actually a separate process from mitosis, and so it initiates before mitosis ends. So technically at this point, cytokinesis is happening in the background. It's started already. I put it at the end because that's when it finishes, but just to be aware, it's actually kind of happening right now already. The cell is starting to cleavage furrow is starting to form there, which just means that it's starting to split the cell into two. Okay, so that's anaphase. That's all that happens. You pull the chromatids apart, X's turn into bananas. And then our last phase here, we've got our DNA on either end of the cell. We've got to put them back in their appropriate package, which is a nucleus. So in telophase, you rebuild the nucleus, and the DNA unwraps itself from its chromatid form and goes back into chromatin, which is just like that loose noodle form. And just to connect this to what we were talking about previously, the reason why you want to decondense it and put it back in its noodle form, where it's just kind of laying about, is because you have to read the DNA to use it. And different sections of the DNA are getting read at the same time. You might have a gene at the top, and a gene in the middle, and a gene towards the end, and a gene at the other end, and they're all being read at the same time. So in order to do that, the DNA has to be laying open so that different parts of it can be accessed at the same time. When it's wound up into chromatids, when it's wound up as a chromatid, you can't access the different parts of the DNA anymore. And so you can't actually use the DNA while you're doing that process. All of the cellular processes that require DNA are on pause while this is happening, while mitosis happens. And then once you unwind it again, then you can start accessing the DNA and using it. It's sort of like you've put all of the pages into a book, and now you're going to like organize your books. While you're doing that, no one can read the books. But when you're done, you can tear out all the pages to your books and throw them out on the floor, and then anyone can just read any page at any time, which is what's happening to the DNA. The pages are being read, like the 5th page and the 26th page and the 80th page might need to be read at the same time. So you just kind of throw all the pages out on the floor so that any enzyme can come and read any particular part at any particular time. Okay, and then lastly, cytokinesis. We split them up. In a lot of eukaryotic cells, this usually takes between 10 and 20 hours, depending on the cell type. So it's relatively long in order for you to actually go through the process. In prokaryotes, this takes less than an hour usually. So prokaryotes can replicate extremely quickly, a lot faster than eukaryotic cells can. Just to show you a corollary to that. Okay. Hopefully, no, I'm not going to get you to do these. Should I? Should I get you to do these? Are these useful? Hold on. Let me just double check here. Oh, no. Actually, I guess you should do this. Yeah. Yeah. Oh, I'm giving you extra work today. That's a bummer. That's a bummer. I wonder, yeah, because, well, you're going to need to know this before, you're going to need to review this before we do the meiosis tomorrow. How can I break that down in a way that isn't so labor intensive for you? Let's see, how is it organized tomorrow? Yeah, there's no way around it. Okay. What I'm going to get you to work on during this block and learning block 3, I want to push some of this stuff into tomorrow, but if I do that, we're going to end up backing stuff out the back of the unit. And the last thing we want is for the unit to overlap and then have you go to your other class and not be done this unit. This will not work out because you're going to be gone for 2 weeks and then you'll come back having probably forgot a lot that we've just done, and then you're going to do a little bit more genetics and then you're going to write your quiz. That is the worst case scenario. So I don't want to do that. These questions shouldn't take a super long time. They're not too bad. They're asking you to describe parts of mitosis, which is a review from last year. So, okay, here's what I need you to do. So I'm going to stop humming and hawing about this and just let you know what you need to do. I'm going to get you to do the protein sequencing practice. Okay, so remember that is on this page right here. Okay, some of you may have already done part of it. It's not super time consuming, assuming that you are comfortable with the process. And if you're not, please stop by the Google Meet. Okay, I'm going to get you to write out the transcription and translation process in your own words. This might take you 20 minutes, okay, or even half an hour is possible. Okay. Shoot, how am I going to work this out so that you have more time to work on this? Because I feel like this is too much. It's all stuff I need you to do, though. Okay. Okay, this is how we'll do it. This is how I'm going to organize it. I'm going to get you to do this activity and summarize transcription and translation. Okay? I also have a... hold on a second here. Oh, there's some extra stuff on here. This is the stuff that's mentioned here in the protein synthesis lesson. And I'm going to go through each of these steps here. A bunch of it is enrichment. Okay, so for example, there's a really cool visualization of what the inside of a cell actually looks like. And it shows what a lot of the proteins involved in transcription and translation actually look like. That's completely enrichment and not necessary for this course. But if you're interested, check out the visualization that's there. If you click on the little link, there's one below here for protein synthesis, which shows you, walks you through what a bunch of these proteins actually look like inside of the cell. It's fascinating. So if you're interested in it, you're welcome to do that. But that's enrichment. Okay, so don't feel like you need to do that. I mentioned the protein sequencing practice page and the transcription and translation process review and writing. Do that. Okay. There is an article here about a recent advancement in using proteins that are based on their amino acid sequence. It's basically using what we just learned about in a practical way. Okay, that is enrichment reading. I don't ask you any questions about that on the quiz, but if you're interested in seeing some of this in action, how we use this knowledge to, do stuff in actual biology, that article is available to you. Okay, but again, don't feel pressure to do that. I realize that I'm pushing barriers on time a little bit here, and so the enrichment activities are exactly that. They're for your interest and enrichment. Okay, there is an exit quiz. Remember that the purpose of these is for you to test yourself on the knowledge that we've just completed and see if you should be going in for extra help or reviewing your notes. You should do that. Okay, I think it's only 5 questions. It's not going to take you a long time, but you should do it. We just went through the meiotic process together. We reviewed mitotic process. But if you did that and you were like, uh-oh, I don't really remember this from last year, I strongly suggest that you watch this video review of mitosis, which will go through the process again. Gives you a little bit of additional detail, but not stuff you necessarily need. And it has some great animations that go along with it as well to help you review mitosis. You really do need to have a firm understanding of mitosis for what we're going to do tomorrow. So if you're not quite there, I recommend this video. If you are there and you're like, yeah, I get mitosis, I remember it from last year, this is not hard for me, then you're good. Okay, you don't need to watch this video. You've definitely got the process at this point. As I mentioned, there are some cell cycle review questions. Okay, I'm actually going to give you time to finish these tomorrow morning after we do attendance. Okay, because I really feel like I've pushed the barrier of time a little bit too much today, and that's not really fair to you. So these cell cycle review questions, I'm going to give you 15 minutes, 20 minutes tomorrow morning. After we take attendance, I'll send you away and you can work on these. As a part of that, and these questions are just about mitosis, okay? As a part of that, the last part here is asking about why do cells divide instead of just getting bigger, which is a great question. And we talked about it in grade 10. If you're in my grade 10 class, I did talk about this last year a little bit. But there's a great reason for that. Why aren't we just giant cells? We're large organisms. Why can't we just be one giant cell? Why do we have to have like a whole bunch of cells? It's so much more complicated to have a bunch of cells. Well, there's a very good reason for that. And in order for you to answer it, I'm going to get you to watch this video. And again, this will be tomorrow morning. You don't feel like you need to do it today. But I'm going to get you to watch this video on surface area to volume ratio and why that specifically is the reason why cells have to divide and they can't just get larger. Okay? So I'm going to do that. I'm going to give you an opportunity to watch that and answer those questions tomorrow morning. Okay? Normally, before we do meiosis, I have you watch this video. It's a summary of meiosis, what the purpose of it is, and in general, what it looks like before we do it together, because it's a very complicated process, and I normally get you to watch it the night before. I'm going to leave this one up to you. If you want to watch this today during Learning Block 3, you can do that. But I will give you time tomorrow morning after we take attendance to watch this video before we get into meiosis. Either way, though, you're going to need to watch it. It is a complicated process, and it very much helps to see animations of these things before we talk about them together. So anything on here anything on day 6 from after this mitosis video, okay, complete cell cycle review questions, that's number 3 and lower, you don't have to do right now, okay? But above number 3, If you need to watch the mitosis video, do it. And completing the questions from protein synthesis and the EXA quiz. Strongly recommend. Okay? So that's where I'm going to cut it, just to make it more manageable for you. Otherwise, I think it's too much work for one day. So I'm going to let you go away. If you have questions, please let me know what they are. I'm happy to review any of this stuff with you. And if you want me to go through any of these processes in more detail right now, please join me during the Google Meet, and I will go over them again with you. Okay, one-on-one, or there might be more than one person in there, but we'll go through them together. Okay, I'm going to stop talking and give you a chance to work. So much to do.